వచ్చింది సార్, వచ్చింది సార్. అలాగే సార్, అలాగే సార్. అదే ఒక్క నిమిషం సార్, అది ఎడిట్ చేస్తున్నాను సార్ కొంచెం. ఎస్ సార్. అలాగే సార్, అలాగే సార్. అంటే కొంచెం ఎడిట్ చేస్తున్నాను సార్, అందుకని సార్. ఓకే సార్. అలాగే సార్.
యూట్యూబ్ లింక్ కాదు, లింక్ పంపించారు.
చెప్పండి.
మీకు మెయిల్ ఓపెన్ చేసుకో.
చూడండి, ఇందులో చూడండి.
ఇక్కడనే మేడం.
ఈమె ఉపవాసం. సో ఏమైనా ఫ్రూట్స్
అవునా? మీకు వచ్చిందా? నాది వచ్చింది ఇలాగా. మీది? మీరు మళ్ళీ ఒకసారి పంపించుకోండి. మెయిల్ వచ్చిందా? ఇప్పుడు వినిపిస్తుంది, ఇదిగోండి. హలో హలో,
పద్మావతి గారు.
వినిపిస్తుందండి.
ఓకే మేడం, ఓకే మేడం.
మీ వాయిస్ వినపడుతుంది సార్ నాకు. నాది వస్తుందా వాయిస్?
మైక్ ఆఫ్ చేయండి మేడం. అందులో మైక్ ఆఫ్ చేయండి. మైక్ ఆఫ్ అయితే పద్మావతి గారు మైక్ ఆఫ్ చేయండి.
నాది ఎలా ఆఫ్ చేయాలి? Okay.
Hello. Hello. Hello.
Sir,
Hi. Hello. This is Tanige Malai.
Hello, sir. Sir, good afternoon.
Hi. Can you hear me, please?
I can hear you, sir.
Yeah. Very
Yeah,
nice. Very
okay.
nice. Yes.
Very good afternoon, sir. Welcome, sir.
Thank you.
All the best, sir.
Yeah. Thank you. So how can I start? Who's going to start first? Hi. Sorry for the interruption. Can you please unmute?
విజయ నిర్మల మేడం ఒకసారి మాట్లాడండి.
Hello. Hello. A very good afternoon to one and all.
Hello.
Hello. May I
Yeah, we can
request-
hear your voice.
Thank you. I request all the participants to unmute your cameras and also video and audio for the smooth conduction of the webinar. Mute.
వస్తుందా వాయిస్?
Hello, we could not hear any audio.
We request all the participants to kindly be patient. We will be starting in a few minutes, and we also request you all to mute your audio. Thank you.
Hello.
Hello,
Sir, good afternoon, sir.
good afternoon.
Okay, sir. ఈ slide ఇలా ఉంచుదామా? అలా ఉంచు. Sir, I'm starting it right now. Hello.
Yeah.
Sir, very good afternoon, sir.
Good afternoon. You can start.
Okay, thank you, sir. Respected Honorable Vice Chancellor, Professor Mokka Jagannatharao garu, Registrar of the University, Professor Gangarao garu, all the EC members of the university, faculty friends, principals of the university, College of Science and Technology, Dr. K. Ramaneshwari, and other principals, deans, and participants. A very good afternoon to you all, and hearty welcome to this international webinar on COVID-19 research strategies and therapeutics. The Department of Life Sciences has organized this, and we hope that all the participants will utilize this opportunity and interact with the eminent research scientists who had been working exclusively in this area. And I think it's the platform to thank the Honorable Vice Chancellor who has facilitated us with this Zoom app and also given the facility. We are thankful to our Vice Chancellor who have been motivating us and always directing us to the post, saying that the lockdown should be taken should be as positive attitude and grab the opportunity to strengthen academically. And I should thank the senior most faculty who had been our advisory, Dr. Mataredi behind this, and Professor K. Sampasivara sir, our mentor, Vice Chancellor of Mizoram, who has guided us. And I thank my faculty friends who have been behind this in organizing this. And I now request our Honorable Vice Chancellor, Mokka Jagannatharao sir, to address his inaugural address to the participants. Before that, a brief introduction about sir. Sir is to tell in a single word, sir is a very good academician who always speaks academically, and he always encourages the faculty and students towards the academy. A man who always encourages in any idea we go with any innovative idea, he's there behind us, and he always never say no to anything that we do for the welfare of the university. With this little introduction, I request the Honorable Vice Chancellor to address the participants.
Thank you, Dr. Vijay Nirmala. And good afternoon, all the participants and resource persons. Today's webinar on COVID-19 research strategies and therapeutics. I congratulate all the organizers. And here we have Principal Dr. Ramaneshwari and my other colleagues, Dr. Matta Reddy, and EC members, Dr. Ramesh, Jagan Mohan Reddy, and Dr. Ashok, and other deans in charge, Registrar, Dr. Y. S. Srinivas Rao, and other principals, deans, and the resource persons I should mention. I'm thankful to them for accepting our invitation to be the speakers today. Dr. Tangamalai Villar, Dr. P. Gopinath, Dr. Buzzi Babu, Dr. Shivakumar, and others. And also, I congratulate all the participants from India and abroad for participating in this webinar. It is a uh, very important topic of the COVID-19 research strategies and the therapeutics. Since the outbreak of COVID-19 in the Wuhan city of China, so we are all now involved in this crisis of COVID, and we are all locked down at your homes, and we do not know still. We are fighting with the enemy, which is invisible. We do not have any clue how we need to contain this virus, this COVID, despite a lot of hard work being done by the scientists and medical doctors and pathologists and microbiologists and others for finding a solution, finding a vaccine. Still, we are all waiting. So being a layman, but being a scientist, but still we have a lot of questions about this virus. A lot of miscommunication initially. The World Health Organization initially said it cannot transmit from human to human. Later, they corrected their statement and they said, okay, it will transmit human to human. By that time, we lost very precious time. If it could have been said early, the Wuhan city could have been just isolated from the rest of the world. So we could have at least contained the transmission to the maximum extent. But it is not the case. It is the Wuhan, from Wuhan, number of people traveled to Italy, traveled to other countries very freely because of this kind of miscommunication. So it's how it happened, we do not know. But still, there are statements that the source is from the bats, from the pangolin, or from other animals. But still, people say it is a questionable source. So these things are making the public confused. We still do not have proper scientific basis from where it is actually it is the genesis is from which place, what is the source. Until you know the source properly, maybe I think getting a vaccine for that may be much more difficult. So that is my idea. Similarly, there are other things to take the precautions from this COVID. We need to use, as per the medical advice, we should keep away at least 6 feet from the person or the persons who are maybe a suspect. We need to have the mask. The mask should cover our mouth and the nose, etc. Then recently, now there is a study from which we know that even this contamination, this infection, may happen through your eyes. So that is a recent study. So this is again another important point where we are getting information time to time. So new information we are getting. So it is clearly an indication that scientists do not have a total knowledge of the occurrence, transmission, infection of this particular virus. For example, being a scientist, they say it is very small. So almost it is 0.125 micron in size. And they say it will survive on various terrestrial environments and different surfaces for 72 hours. And if you're 6 feet away, it is okay. But my question as a scientist, if it is so small, is of 0.125 microns, can't we expect that it is airborne? It will travel in air for further distances during this 72-hour period. So that is, as a layman, that is my question. Similarly, we see in a number of cases, a plane is originated in a given country, X, where all the passengers are tested for the corona, and when they are negative, they are allowed to board. by the time they reach the destination, at least 5% or 6% people are contaminated. So definitely we see there is some sources within the aircraft. This machine is carrying the virus. So despite the sanitization or disinfection of these aircrafts, but still we need to believe this virus is hanging around in various parts of the aircraft, maybe including its seats or roof or ducts of air conditioning or the lavatories or toilets, etc. So in this way, still there is a lot of questions to be answered by the experts about the origin, about the transmission, about the contamination, about the infection and survival, and how many days it will survive on different surfaces, etc., etc. This is about these questions are asked by laymen to these experts. But another important thing, as webinar states, we are all working on finding a vaccine. The vaccine needs to be developed immediately. We are all waiting to see that day. That is the day the entire world is looking at these scientists, these medical doctors, these microbiologists. We are looking at you so that you can get all of us out of this danger. So the entire world is now suffering. Is it financially, industrially, economically, and education-wise, and socially? Many people are struggling, especially the day-to-day breadwinners and day-to-day laborers. They lost their livelihood and they are staying at homes because of this invisible enemy. So we look forward, and also we wonder when the medical technology has this kind of advancement, so why it is taking such a long time to develop a vaccine? So maybe there are a lot of limitations, but still we need to work out a plan, a strategy for future, because such pandemics may be occurring in future as well. So this should be a big lesson for us to be well prepared for any future eventuality of such pandemics. There should be a continuous, at global level, research continuously should be happening to be ready with the possible pandemics in future. It is not like once a pandemic happens, then we start working on a given vaccine. So I strongly feel there should be a strategic global plan of all the scientists and the scientific laboratories working on such viruses and vaccines. They should have a plan so that any eventuality, any future pandemics can be tackled in a shorter time period, so that the people are protected, the world is protected. So in this way, there may be different therapeutic techniques, technologies, maybe a combination of them can be used to understand, maybe to develop a vaccine, a better vaccine in a shorter period of time. So I wish all the deliberations today will be focusing on these aspects, and definitely we'll come up with good uh, deliberations and good outcome out of this webinar. Once again, I congratulate all the organizers for giving me this opportunity, and I congratulate all the participants for participating in this webinar. Being the Vice Chancellor of Adikavi Nannaya University, I want to say that all our faculty, from sciences, from engineering, and from arts, commerce, and humanities, they are very active during this lockdown period and continuously involved in various academic activities. And we look forward for many webinars in future. Thank you very much. Thank you.
జనరల్గా ఏంటంటే అగ్రికల్చర్ సీజన్ దాటగానే మోస్ట్ ఆఫ్ ది పీపుల్.
Thank you, Dr. Vijay Namala.
Thank you, sir, for your valuable message. Sir, really, we thank you for the detailed information that you have given us. I think all the participants have got enlightened before the speakers itself has spoken. Our sir has enlightened you about the origin, transmission, kind of you alerting all of us about the important thing that in future many pandemic situations may come, and we all should be well prepared to face such kind of situation. So it is really, sir, a very good awareness that you have created among our participants and all of us. Thank you very much, sir, for your for inaugurating the international webinar. We hope that the vaccine or drug may come very soon in future, and as you wished. Thank you so much, sir, for your support and for your inaugural address.
Thank you.
Thank you, sir. Now I request our faculty, Dr. Murli Mohan, to introduce our speaker, the first speaker, Dr. Tangamalai, sir.
We could not hear you very well. Please, can you increase the volume?
Am I audible?
Not very much.
Basically, he has completed his post graduation from Bharatiya University. Is it fine, sir, now?
It's not audible, please.
Sir, am I audible? Am I audible?
No, not very much.
Okay, yeah, that's one second. Okay, can I go ahead right now? Hello, is it fine now? Yeah, hello. Thank you. So, okay, it gives me great pleasure to introduce Dr. Tangamalai Pellayar. He has completed his post-graduation from Bharatiya University, and he did his PhD work from Chungam National University on synthetic medicinal chemistry. Dr. Pellayar is he has several contributions, and he has published. Right now, he is working as a senior scientist on G protein. Right now, he has a couple working as senior scientist at University of Bonn, Germany. he has published several research articles in peer-reviewed journals, and he is also acting as a reviewer for several publications, including a Nature Publishing Group elsewhere, Springer, and several other important publishing groups across the world. And he is involved in several research projects on cancer, muscular dystrophy, and neurodegenerative diseases, etc. Apart from this, he has participated in many international conferences and workshops. Dr. Pelliar, his research interest is on synthetic medicinal chemistry and development of drugs with potential biological applications. In addition, he has several awards and recognition to his credit, and mainly the Humboldt Bayer Research Award. For this purpose, this is for his postdoctoral study from University of Bonn, Germany. This is one of the prestigious awards to his credit, and National Research Foundation, and there is one more scholarship from National Research Foundation of Korea for the graduate study from South Korea. So as mentioned earlier, he is right now working as senior scientist at Pharmaceutical Institute, University of Bonn, Germany, where he is right now co-supervising doctoral and master students in parallel. So with this brief introduction, I would like to invite Dr. Thangamali Pelliar to this great platform where he will be contributing from his talk and he is going to talk on drug development in medicinal chemistry researches towards SARS-CoV-2 and COVID-19 therapeutics. Thank you, and sir, the floor is yours right now. Thank you.
Hello.
Yes, audible, sir.
Yes, very nice. Yeah. Good afternoon to everybody. My name is Thangamali Pelliar. I'm working as a senior scientist at the Pharmaceutical Institute, University of Bonn in Germany. I would like to thank the committee members for giving me this opportunity. I'm really happy to share some of my research related to the SARS and coronaviruses. So to say exactly the topic about my talk is drug development and medicinal chemistry researchers toward the SARS coronaviruses and COVID-19 therapeutics. Yeah, please let me know if I could change, if I need to change something.
I think you can give full screen because it is.
Yeah, I'm doing it. Yeah. That's fine now, right?
Yeah. Thank you very much, sir.
Yes, good.
Thank you, sir.
So let me continue. So the brief content of my talk is I'm going to introduce about the coronaviruses, what is coronaviruses, and their taxonomy, structure, how they infect from the animal, and their infection cycle, therapeutic strategies, and finally the conclusions. As Vice Chancellor mentioned, that we are in a very difficult situation now because of the new outbreak. But I would like to insist that this is not the first time that we are suffering from the coronaviruses. Since 1920s, we have discovered about 7 human coronaviruses. I would like to insist that the first 4 coronaviruses are responsible for the 1/3 common cold in humans globally. Well, since the beginning of 21st century, 3 human coronaviruses crossed species barrier to infect humans. And these coronaviruses are severe acute respiratory syndrome, Middle East respiratory syndrome, and the newest one which we are suffering from. They all belongs to beta coronaviruses. Let me explain later how they call it as beta coronaviruses. So the first outbreak was happened in 2002, of course, in China. It affected more than 8,000 people with a fatality rate of 10 to 15%. 10 to 15%. Exactly 10 years later, there is another virus originated from the Middle East, was affected more than 1,400 people with a mortality rate of 39% worldwide. And now we are suffering from another virus. It is not surprised that it is also originated from the China. So I think they have very close relationship with, with the human coronaviruses. So as of June 5th, it infected more than 6 million people around the planet and claimed more than 380,000 lives, and it is still continuously infecting. We don't know when it will stop. Very unfortunately, we have no approved drugs or vaccine for any human coronavirus related disease. In order to do something, it is very, very important to understand what is human coronaviruses and their infections and their life cycle in order to develop a potent therapeutic targets. Okay, I said in the beginning that what we are facing the coronavirus is belong to the beta coronaviruses. How they classify the virus? Well, according to the viruses are classified according to the International Committee for Taxonomy of Viruses. Well, the human coronaviruses belongs to two subfamily that are coronavirinae and toravirinae under the family of coronaviridae, which in turn is formed under the order nidovirales. The subfamily coronavirinae is further classified into 4 main genera, that is alpha, beta, gamma, delta coronaviruses. As you see here, hello? Sorry, I think there was some interruption, so let me continue. So there are 4 genera of coronaviruses. As you can see here, the very highly dangerous coronaviruses are highlighted in red color, which all belong to the beta coronaviruses. So the research says that both alpha and beta coronaviruses infect only humans, while gamma and delta coronaviruses infect birds and animals. Well, how they infect? The next question is, we know what is coronaviruses, but how does it looks like? Yeah, how does it looks like? Actually, the coronavirus is a positive sense single-stranded RNA viruses with the largest size of genome in the range of 29 to 32 kilobytes. It has 4 structural proteins like spike glycoproteins, which will be useful for infecting the receptor, membrane protein, envelope protein, and nucleoprotein. This virus encodes for 2 polyproteins, which can be further cleaved by the proteases to produce the viral particles. I will give you very detail about this part in coming slides. Well, how they infect and where they come from, the human coronaviruses. The research says that all human coronaviruses have animal origins, most probably bats and rodents, and there are small animals like cattle, mice. Yes, so you can see me now. They have the animal origins. Well, the coronaviruses are associating with a lot of animals, and it has a very good relationship with animals since thousands and billions of even millions of years, even even before the human comes to the planet. And they both benefit each other by associating. And it is very clear that the RNA coronavirus need a host to replicate its new virus and new generations. When you are disturbing this relationship, that the virus jumps from animal to another new animal, that is human. Yeah. So there are different kind of human coronaviruses, and they use different cellular to infect humans, and they have different hosts. And it is very important that how we are disturbing the nature, or how we are invading the nature. It is really important to think about whether we are going beyond the nature. Yeah. So once it's find a suitable partner in humans, then it will begin the life cycle. So as you see there, this is a typical coronavirus infection cycle. I'm just giving example for the new SARS coronavirus 2. Well, the virus begins its life cycle from the entry by recognizing a suitable receptor. In this case of SARS-2, it is angiotensin converting enzyme, which is very similar to the case of SARS coronavirus 1. Upon its entry, the virus is decoded to produce the viral mRNA, which is readily translated to polyproteins. And these polyproteins can be cleaved by proteases to different structural, non-structural functional proteins, and RNA polymerase and RNA dependent and RNA polymerases. Once the virus particles assemble together, and it will mature and exotized to another cell for infecting new cells. So these are the 4 major steps in coronavirus's life cycle. So when you interrupt any stage of virus replications, this will become an important therapeutic strategy. According to the recent research report, it says that there is more than 60 druggable targets are available to target, each of which has multiple binding sites. But I would say 4 important targets are: the first is viral entry, and the second one is proteases. The proteases are divided into 2 parts. I will give you explanation in the coming slides. And RNA dependent and RNA polymerase. These 4 targets are considered as very important, and huge research efforts have been taken so far to combat the coronavirus related disease. Okay. The first step is the viral entry. Of course, the viral entry is the first step. Once the virus finds a suitable partner, in this case of SARS-1 and SARS-2, it's angiotensin converting enzyme. It recognizes the receptor. I would like to insist that even the virus uses its spike protein, which is divided into 2 parts. One is S1 receptor S1, another one is S2. The S1 is important for the receptor binding, and S2 important for the virus fusion into the host cellular membrane. So when you are targeting, here we have 2 options to target. One is virus-based and another one is host-based. The virus-based means you can target the spike protein. You can develop the anti-spike monoclonal antibodies or vaccines. But on the other hand, you can target the human receptor, that is angiotensin converting enzyme 2. So these are 2 options. Of course, there are very complicated process. The virus entry is not a simple one. There are so many mechanisms are involving in this. But in order to better understanding these are the simplest way to explain how can we target the virus entry. So whatever we are doing the research is based on the experience what we had for the SARS coronavirus one, which was in 2010. Huge research efforts have been taken for this virus, even though we could not able to find something which can be treated, which can treat the new virus. But we have a lot of background and research efforts for already for the SARS-1 viruses. As you see here, both it is clear that both SARS-1 and SARS-2 are behaving very similarly, and they use very similar receptor to infect the humans. As you see here, I'm just comparing the SARS-2 spike protein and the SARS-1 spike protein, and they look like very similar, even for the sub-1 and sub-2. So what the researcher done for SARS-1 coronaviruses, they screened a lot of natural product and synthetic compounds from various sources, and they could be able to find some drugs like compound that could selectively inhibit the S1 subunit of SARS-1 spike protein. In this very interesting drug is imatinib, which is already approved for for treating the cancer, but we have no information about whether it has been investigated already for SARS-2, I mean the COVID-19 disease. We have no information. The next one is virus entry, which is targeting angiotensin converting enzyme. And this is just a three-dimensional picture that how the spike protein interact with the AC2. As I mentioned earlier that they both use SARS-1 and SARS-2 both use angiotensin converting enzyme, and they have very similar binding mode, and the receptor is highly expressed in the lung. That's why we get the respiratory syndrome. So when you target these interactions, I mean, when you target the spike protein and AC2 interaction, then this will become an important therapeutic approach for developing a novel drug. So based on the experience that we had for SARS-1 outbreak, we have screened and tested so many drugs and experimental compound, whether if they could have some effect on the virus entry, whether we could block the viral entry. So among that, these 4 drugs are very interesting, which are always you can see in the news, and especially the chloroquine, hydroxychloroquine, they become very famous, and it proposed by many scientists that it can be a possible treatment option for COVID. And it becomes also controversial when it was proposed by a politician. Yeah, of course, you know who's that, because he wanted to advertise this, the chloroquine. Since then, and even the World Health Organization initially declared that chloroquine was one of the drugs among 4 was included for testing the possibility of therapeutic options. Since then, around the world, there are so many research organizations and companies, they conducted clinical trials, and until today, they did not find any good benefit. They did not find a benefit of chloroquine and hydroxychloroquine. While when conducting the experiment, many people were died. So it is no more recommended. I mean, the hydroxychloroquine and the chloroquines are no more recommended for treating option for the COVID. There is another drug which is called umifinovir. It's like a broad spectral antiviral compound, which is blocking the viral entry, inhibiting the virus host cell fusions. But it is being currently investigated in a clinical trial for the treatment of COVID-19, but we have no update so far what happened about the results. Probably we will hear sooner or later about this treatment. Another interesting compound is camostat. It's a serine protease inhibitor to block the viral entry, and which is also reduce the reduce the infections of lung cells by SARS coronaviruses too. Well, this compound is already approved in Japan for the treatment of chronic pancreatic associated pain, and hopefully we will hear something good about this compound as well, this drug as well. Yeah, another important thing to address that once the scientists discovered that angiotensin converting enzyme 2 is a receptor for SARS coronavirus 2, there was a rumor and speculations emerged about potentially negative consequences of angiotensin inhibitors and blockers therapy in COVID-19 patients. They claim, some scientists, they claim the expressions of angiotensin converting enzyme 2 increases in patients who are suffering with cardiovascular disease. Sorry. They also claimed the people who are taking ACE2 drugs, increasing drugs, would be at higher risk for the COVID-19 because they think that the treatment with angiotensin inhibitors, converting enzyme inhibitors and blockers would raise a level of AC2. I think they predicted the mechanism is like that the SARS can easily recognize the AC2 for on the people who are suffering from cardiovascular disease, because they claim that the expressions of angiotensin converting enzyme will be higher when compared to the normal people. But it is not the case. It is not the case because many experimental data show that this drug can be a protective rather than the harmful, because the protective arm of the renin angiotensin system is really important for the people who suffer, or for the people who are suffering from the cardiovascular disease, and this could be a novel strategy for the pulmonary disease. So what I want to say here, there should be no panic about these drugs which are already taken by the people, but there was always miscommunications and rumors and about these drugs that it should not be taken, because these drugs are really, really important to save millions of people who are suffering from the cardiovascular disease. Okay, this is about the viral entry. Viral entry. The next topic is proteases. Proteases are really, really important. Proteases are really, really important because they cleaves the polyproteins which are produced by viral RNA, and they cleaves into many non-structural functional proteins and the structural proteins here, like envelope, membrane, nuclear protein, etc. So when you are targeting these two proteases, this will become also an important approach to control the viral replications, especially main protease. So let me say something about the papain-like proteases inhibitors. We have not studied much about these inhibitors when compared to main proteases, because it cleaves only 3 non-structural proteins. When compared to main protease, this is not that important, though it is as one of the important target for controlling the viral replications. They have Professor Arun Ghosh reported some compounds regarding the naphthalene-based scaffold, and which showed some activity against the SARS coronavirus 1. And also they tested some compounds, SARS coronavirus 1, and they show some activity, but really there is no good chance to develop a drug based on this target. And recently they identified some approved drugs like thiopurine and 6-thioguanine, whether it can be possible for the treatment option for the SARS coronaviruses and MERS coronaviruses. But there is no report for SARS coronavirus 2, because these are the anti-cancer drug, I think. So which could be obviously a toxic to the cells. I think this could be the reason why they did not continue, or they did not investigate for the SARS-2 virus. Another important target is main protease. Yeah, this is one of the very important target among coronaviruses because it cleaves 11 non-structural protease, which are highly important for the viral replications. So once you target this enzyme, you could able to control the viral replications. And the most important thing is the main protease is highly conserved among all coronaviruses. So if you can target the main proteases of SARS, then there is a possibility of developing broad spectral antiviral compound, which can be used for the current outbreak as well as for the future outbreak. And this is the structure which shows the conservation of active sites, substrate binding pocket of 12 coronaviruses. And basically it is a dimer, homodimer, which has 3 domains, and the substrate binds between domain 1 and domain 2. The dimerization is really, really important for the catalytic activity of these proteases. Okay. So what about the therapeutics targeting SARS coronavirus 1 and 2 main proteases? Mostly the inhibitors of main proteases are classified into two types. One is peptide based, another one is small molecule based. Peptide based inhibitors are always derived from its natural substrate sequence, which has high conservations, and this red arrow mark indicates the cleavage site by the main protease. And whatever you have the amino acid residue on the left part, we used to name it like P1, P2, P indicates like pockets, yeah, P1, and we numbering like P1, P2, P3, P4. And on the right side, we just used to name it like P prime. So what we did, we developed a tripeptide based on the natural substrate sequence. Actually, there is a theory that a good substrate can become a good inhibitor by modifying some part of the natural substrates. So what we did, we took exactly P1, P2, P3, from P1 to P3, exactly what we have in the natural sequence, and then we developed some drug. And this is one of the best one in the beginning, which showed about 2.2 micromolar. But which is not enough. So we wanted to improve the activity. We docked this compound as I shown here, and we were looking for, is there any opportunity to improve the activity? And there is an interesting point that we found here, that there is a space, and we thought that maybe if you fill this space, probably we could increase the activity. Accordingly, we took this as a lead compound and synthesized so many derivatives here. And we found some best residues, which showed really 3 nanomolar inhibiting the SARS-1 main pro activity. But the problem is, this is a typical peptide, and which may lack the cell penetrations and other drug-like properties. So what we did, we wanted to reduce the peptidic nature. In order to do that, we just removed the one amino acids in the inhibitor from tripeptide to dipeptide, and we studied its structure activity relationship. We found some interesting moieties, which is connecting here. They showed some moderate activity, and one of the best one was like 0.39 micromolar. And we docked this compound, and we found that there is an interesting hydrogen bond interactions with a backbone amino acids, glutamic acid. And according to several studies and comparing the active sites of main proteases, if you're able to make a compound which can interact with backbone amino acids, you may have a chance to increase the activity rapidly when compared to targeting other amino acid residues. And And also it will be good for making broad spectral antiviral compound because they are highly conserved, especially the backbone amino acids. But as you see here, this activity is not enough. So we wanted to further improve the activity, and we had an idea that to rigidify these inhibitors to show something. And we have investigated with many different cyclic structure, and we found indole was one of the best scaffold which could able to fill here, effectively. And also this rigidification helps to improve and make additional hydrogen bond interaction with other amino acids. And we were really glad that we could able to maintain the activity of dipeptide, which is very similar to a tripeptide, which I showed earlier. And the compound shows us like almost 6 nanomolar. The advantage of this molecule is we reduce the molecular size very much, and this compound almost looks like a small molecules. And we also calculated the CELAC p value is really good. And now we are testing this compound for the new virus, SARS coronavirus 2, and we got already some activity which I'm not allowed to show here, but which is already promising for the SARS coronavirus 2. And the further studies will be continued with the cell-based bioassay and other studies like pharmacokinetics, staticity, and so on. This is one from basically from my work. Yes, and then another one is in the beginning of this year, Shang et al. Reported a series of ketoamide inhibitors, which was basically reported for SARS coronavirus, and he wanted to improve the activity for the new virus, and he sequentially modified according to the activity against SARS-2 main pro. They were able to get very good compound here, as I shown here, compound C, and they immediately crystallized here, and you see that the compound is beautifully binding to the enzymes. They have tested this compound on various cell culture and animal studies, and this compound really show very good clinical profile. I don't know what is the current state of this compound now. If they are testing already in a human, the animals or something like this. But this is one of the very potent candidate that has been already developed for COVID-2. We should be considered to test more. In parallel, in the same year, there is another group from China. They discovered a series of already approved drugs or the experimental drugs. Hello? Hello.
Hello.
Yeah, very good. Yes. Now it's fine.
Hello.
Yeah. Yes, let me continue, please. So in parallel, there was a series of drugs which were reported for SARS-2 main pro inhibitory activity. For example, sinanserin, disulfiram, chemophore, which is the antineoplastic agent, epsilon, which is well established in terms of toxicity and also for other activities, and chinconin, and so on. As mechanistically, they just modify the target by making the covalent bond. Well, it is questionable always whether the covalent bond inhibitors are good for developing as drug, because it may increase the off-target activity that leads to the toxicity. That's one question. But now the very important thing is to find something best, something better inhibitor for SARS coronavirus 2. And later we can think about the toxicity and so many things. This is very important study in the beginning of this year after the outbreak of COVID-19. And the epsilon shows very good cell-based bioassay activity with EC50 of 4.6 micromolar, and it was reported to reduce the viral infections in the cells. And they proposed that it can be tested in animal studies, but we do not know what is going on right now with this compound. Well, and also other proteases inhibitors which are reported for the HIV virus also tested for the COVID-19. The two important examples are lopinavir and ritonavir. Especially the combination, the scientists expected that the combination would work more effectively instead of testing separate of each of them. And also, it's important to mention that both drugs were already reported for the SARS-1 and MERS coronaviruses. There was some preclinical experimental data, preclinical experimental research and studies, but so far no benefits was observed based on these two compounds, either by alone or the combinations. And as the World Health Organization also included these combinations in the international solidarity trial, but there is still some clinical trials are going on with patients, but there is no update so far, but we could expect to hear something as soon as possible. Yes, the fourth important target is RNA-dependent RNA polymerase. As I highlighted here, this RNA-dependent and RNA polymerase is highly crucial for duplicating the viral mRNA, which can be used infect the another cells. So this is really important. There was already reported like remdesivir, favipiravir for targeting the RNA polymerase enzymes. These are some selected examples that have been reported in the literature, and among them, remdesivir and favipiravir attracted much attention because they show very promising activity in controlling the viral replications. Well, what is the mechanism of remdesivir? The remdesivir is a prodrug. Once it is in the body, the amino acid can be cleaved and triphosphate can be added to the remdesivir. Then it can bind into the primer of the RNA dependent and RNA polymerase. With that will lead to complete inhibit the polymerization activity. That's the basic mechanism of this compound. Another important thing is this enzyme, as like main protease, is highly conserved, and it is important for all RNA viruses. So this is also one of the very important enzyme to target to combat the RNA virus related diseases. There have been so many studies about this compound, so many studies, so many news, and so many promotion about this compound. Still, many clinical trials are going on, but it is always gives a mixed results. One study says it's really very good, and one study says it's not good. So I don't know whether they make us to confuse or what is going behind it. But that is the only one right now, the current hope with the high hope that it could become a drug. Basically, this compound was this drug was developed for the Ebola and Mark virus. And remdesivir also reported to inhibit the SARS-1 and MERS coronaviruses. But once the new outbreak was started, many scientists were already taken this compound very seriously to test clinically. But there have been huge clinical trials are going on, and you get always some mixed results, as I mentioned. And we are waiting to hear something consistent and positive results from the studies. But still, we are not clear about this one. So as a conclusion, the coronavirus outbreak is not the first time. It is being with us, and it will be being with us. So we have to be ready with drugs or vaccines for the future outbreak. But unfortunately, we have no approved drugs and vaccines so far for any human corona-related diseases. As I discussed throughout the presentations, the viral entry proteases like papain-like protease and main protease and RNA-dependent and RNA polymerases are very important target. Especially main protease and RNA-dependent RNA polymerases are highly conserved. So you will have a chance to develop broad spectral antiviral compounds when you're targeting these two enzymes. And more importantly, the people who are taking the angiotensin inhibitors or angiotensin receptor blockers, they do not have to afraid if they suffer from the COVID. So it doesn't do anything for them because it has a very protective effect rather than the harmful effect. So there should be no panic. So finally, I would like to thank you all for listening. And I hope you could you learn something. And I'm very welcome to if you ask some questions. And at the end, please keep washing your hand, wear mask, and keep distance, and save you as well as other people. Thank you so much for your kind attention. If you have any questions, please welcome. Hello?
Sir?
Yes.
Sir, thank you very much, sir, for your presentation. And
Yes.
we have some questions like in the chat box, we have seen some questions like, please elaborate the plasma therapy for COVID-19 and the precautionary measures for it. One question from the participant.
Please?
Please elaborate the plasma therapy
Okay.
for COVID-19 and the precautionary measures for it.
I'm not very familiar with the plasma therapy actually.
Sir, another question. Can't we model a drug as hydroxychloroquine, drug increasing endoplasmal pH by using bioinformatics?
Yeah, that's actually the basic mechanism of hydroxychloroquine and chloroquine are increasing the pH of endosomal. Of course, it's true, but they have to propose what is meant by the bioinformatics. The one who asked, they have to propose the model or concept. How can you increase the pH of endosomal by using the bioinformatics? This is what I wanted to say.
Hello.
Hello.
Hello. So another question.
Yeah.
That the Indians are using in the sense it can prevent you or it works well. Can you tell the exact name of the drug?
Which one?
So you have mentioned some medicinal drugs in your presentation. So
yeah.
During against the COVID-19, is there any particular combination that we're using, especially in India?
The audio is not very clear, actually. Yeah.
Hello.
Hello. Yeah. I can hear now very well. Yes.
Yeah. So in India, so whatever we're using that hydroxychloroquine or with combination of these drugs we're using, can you tell the exact combination or particular drug that working well, especially in India?
First, what I want to say, there is no particular drug that can be used to treat this coronavirus-related disease for any coronaviruses. But scientists were trying randomly but with many drugs, yeah, based on the previous experience, and it is always depends on the patient how they tolerate to the drug.
Okay. And there is a difference of these drugs activity between China and Indian conditions. This utility of these drugs between China and India, there will be a difference of activity of that working of this drug.
Yeah, that is very clear. It is because of the ethnicity difference. You know, people have different the immune response to the drug.
Okay. Thank you.
It is depends on the people, I think, because there is no such a rational concept for those drugs, because this drugs works on some people very well, and some people they kill them because of the immune response, I think.
Okay, that depends. Yeah. Okay.
Yes.
Thank you. Thank you.
Yeah.
Thank you.
Yeah.
Thank you.
Is there more, please?
So another question from the participant.
Yeah.
Amulya, can this virus cross placental barrier?
Cross placental barrier. Yes, it depends if the virus could find the receptor, okay, in such a system, and why not?
Okay.
Because so far the receptor which is expressing on lung cells, yeah, this mainly affecting the lungs. If there is a receptor which can recognize by virus in the placenta, of course it is possible. There is no limit, I think.
Thank you, sir.
Yeah.
Another question, sir.
Yeah, please.
What about eye contact, sir? Is it there is a viral like it's transmitted through eye contact? Another question.
It is. I think it is possible in all ways, and it is also questionable whether it will give you harmful effect, harmful effect or not, because the virus infection is based on your immune response. Yeah, if some people if your immune response is really very strong, your immune system could simply handle the virus and can kill it. But the people who are suffering from the immune response, they can even easily infected by this a normal cold.
So last question now. It is the last question.
Yeah.
If a COVID recovered patient conceives, will the new baby carry COVID genes? Hello.
I think yeah.
Am I audible, sir?
I'm not a doctor to say like that, but it is. I think it is possible. I think.
Okay.
I think it is possible.
Thank you, sir.
Yeah.
Thank you, sir.
Okay.
For your presentation and sparing your valuable time for enlightening our participants. Thanks a lot, sir.
Yes, thank you for inviting me again, and I wish all other speakers good luck. And yeah, thank you very much.
We look forward for your support in future also.
Yes,
sir.
yes. Thank you so much, sir. Thank you. Yeah, bye.
Bye, sir. Thank you. Sir.
Bye.
Screen sharing. I think
I can remove now.
Yeah. Now our next Our next speaker is, hello, Which Babu, hello.
Hello. Yes, ma'am. I'm on meeting.
Welcome. I think you are there. Now I request our faculty member to introduce Dr. Padmavati to introduce Dr. Satish to introduce Bujji Babu to the participants.
So good morning, all of you. It's a great pleasure to me to introduce the mute. Hello, good afternoon, all of you. So it's a great pleasure to me to introduce Dr. C. Bujji Babu, who is addressing the topic cardiac dysfunctions of coronavirus natural therapy. Dr. Bujji Babu presently working as postdoctoral scholar at University of South Florida, United States of America. He did his MSc in zoology from Adikavi Nannaya University, Rajahmundry. Later, he did PhD from Chungnam National University, South Korea. Dr. Bujji Babu's research interests are in cardiac electrophysiology, whole genome sequence analysis, computational biology, molecular biology, etc. Dr. Bujji Babu received many awards and fellowships. To mention a few are Outstanding Postdoctoral Award from University of South Florida, USA, American Heart Association Postdoctoral Award, University of South Florida, USA, and Academic Excellence Award from Chungnam National University, South Korea, and Best Poster Presentation Award from Adikavi Nannaya University, India. He published several articles and peer-reviewed journals. And with this brief introduction, now I invite Dr. Bujji Babu to deliver his talk.
Thank you very much for a great introduction. First of all, my hi to Honorable Vice Chancellor Jagannath Rao Garu and Register Ganga Rao Garu, Principal Dr. Ramaneswari Garu, and Organizer Dr. Vijnabhana Garu, and all the faculty members at Adikavi University. And I'm feeling very happy, and I'm feeling such a honor to be a presenter as an Adikavi University graduate student. Hi to everybody. Hope you're doing good and healthy with your loved ones. Before I jump into my topic, so hope you guys see my screen. So how about now?
Bujji, you can go ahead.
Okay. Okay, thank you very much for confirmation. So today my topic title is cardiac dysfunction of coronavirus. So as he mentioned, like my major research is cardiac electrophysiology. So for the past 3 years, I'm working on chloroquine, and chloroquine is a small molecule which is blocking IKC channel or potassium channel, preventing atrial fibrillation. So for the reason, so I'm recently interested designing or writing reviews which are related to the chloroquine compounds and which can prevent COVID-19 more powerful than non-hydroxychloroquine. Well, recently there are many studies proved that in the COVID-19, they found many cardiac dysfunctions, including hypertrophy, cardiomyopathy, arrhythmia, and also like myocardial infarctions. So after I heard like Tangamalai's presentation, I deleted a few slides because he addressed all the introduction, but I still keep some because it's some introduction required for the presentation. So in this topic, I'm going to talk a little bit about introduction, pandemic, and structure, mechanisms, and mainly cardiac dysfunction results in coronavirus or during the treatment of coronavirus. So I think hide this. Good. So SARS is like a severe acute respiratory syndrome. SARS is a viral respiratory illness caused by coronavirus called SARS-associated coronavirus. SARS was first reported in Asia in February 2003. Okay, great. So in this slide, how the SARS virus originated in animals and how it's transmitting to the humans via animals. So there are many viruses originated in animals and it's transmitted to the animals. But some coronavirus, there are 4 coronaviruses, which is like SARS coronavirus and SARS coronavirus, MERS coronavirus, and SARS coronavirus 2. And among these 4, 3 are like transmitting through animals and originated in animals. So SARS COVID-19 and MERS COVID and SARS COVID-1 about these 3 and Dr. Thangamala addressed and how the mechanisms and the introduction. And coming to the pandemic is an epidemic of an infectious disease that has spread across the world or in the big place. So coming to the COVID-19, which is originator or started in China, Wuhan city in December 2019. After that, it spread very rapidly around the world. So that's the reason the World Organization on March 11, 2020, they announced COVID-19 was declared as a pandemic by World Organization. So coming to the cases, it's affected all the world in a very large number. So far, 6.4 million people are confirmed positive cases with this coronavirus. And coming to the statistics, and recently and every day, this number is like increasing. On June 4th, like recently, the positive cases are 1,29,000. So far, 3,82,000 people are sacrificed their lives because of this coronavirus. If you see this map, if you Google it, just like COVID-19, and you can see like big map, like how this virus is spreading over the world and very rapidly, and how many people are affected, and how dangerous this is, and how speedily spreading. So if I go to structural evidences, the structure, coronavirus is like crown-like structure, and it has several proteins, in case like a spike-like protein, which is the main target to reach the host cell, and it has M protein, E protein, and it has envelope. Even inside, there is RNA and protein structural, which can enter into the cells and then replicate and produce several viruses. And that's the reason it affect and kill the cells. And that's in scientific manner, it's called like apoptosis or cellular death. Coming to the transmission route is involving combination of hands and surface in direct contact. And here I just find like 4 ways. First, if any infected individual sneeze or cough, the droplets spread into the air. And if it touches directly the person with his hands or whatever it is, and it can infect with another uninfected individual. Or there are chances to travel or like infect airborne, because there are many studies recently published like airborne. It can be travel like 24 feet, 6 feet, or like 10 feet. So there is no consistent, but still it's considered as airborne. And also there is indirect way, like if uninfected individual use infected individuals, things like clothes or cards or bats, whatever it is, there are chances to infect. So this slide is necessary to tell our audience, because this is very important. Coming to the Okay. So how this is going to be infected? So this coronavirus having spike-like structure is a protein, and it has a receptor binding domain. With this domain, it's reached to the AC receptor in the host cell, and it can enter into the human cells or lung cells, and it diffuse the cellular virus membrane, and the RNA released into the cytosol. And after this process, the translational and it replicates the RNAs. The replicated RNAs goes to the endoplasmic reticulum and Golgi complex and provides the S protein, E protein, M protein as a sheet, and this replicates can hold as a sleeve. And this one can go out as a exocytosis manner. And finally, this kills the cells. It is called like a cellular death or apoptosis. So what happened when it's affected? For example, immune response during SARS-CoV infection. So here, if the SARS-CoV inhale or enter into the lungs or into the body, and here you can see the alveolus is clear alveolus. When the virus enter, it can destroy all the alveolus and lungs. And here is the insert is a CAT scan picture from COVID-19 infected patients. In a very early stage, this picture is taken. Here you can see the left lung, right lung, and heart. In the right lung, you can see the arrow marks where it's infected and it's spread to the other place. And this is in the very beginning stage and can lead to all the lungs, and it can destroy the body and it can destroy the immune system. So what happened in general way? So this lungs is destroying the lung cells, and lungs are affecting. So lungs function is giving the oxygen and oxygen to the blood, and blood carries oxygen to the body parts. Ultimately, it causes the hypoxia and several other cellular physiological modulations, and finally body collapsing with their daily functions and all in general way. Coming to the cardiac function, so this is my main major concerns, like cardiac dysfunctions resulting in the coronavirus or during the treatment of coronavirus. So if you come to the normal cardiac electrophysiology, so normal electrophysiology, heartbeats origin at SC node, and that's electrical signals travel to the AV node, and from the AV node, it can go through the heart via the Purkinje fibers. And in this insert, you can see the ECG, and there are two heart rate, two beats. And here, the small peak is called P wave, and this is QRS. And hope you are seeing my cursor. Okay, so and this is the T wave. So in the normal heartbeat, it's beat generally, and like how it's organizing the signals, electrical signal as SA node, and it's traveling to the AV node, and it can spread all the world, out of the heart. And finally, if it is not normal, like it is beat slow, it is called like bradycardia, and if it is beat fast, it's called tachycardia. It is also called like arrhythmia. Coming to the myocardial injury, so there are many reports done in China, especially in the National Health Commission. China reported that almost 12% patients without non-CVD had evaluated troponin levels of cardiac arrest during the hospitalization. And in one study, they choose like 138 hospitalized patients with COVID-19 in Wuhan City, China, and they found like cardiac injury. And in the patients, they found elevated levels of troponin I, and also they found abnormal ECG and echocardiograph graphs. And in the patients, and they quantified these troponin 1 levels, and they found 8.8 picograms per ml versus 2.5 picograms for survivors. That means in non-survivors have 8.8 picograms. And they continuously tested this troponin I levels in this case, and they found survivors did not change significant effect. And in this case, they found like 24.7 picograms per ml on day 7 to 55.7 picograms. And the day 13, they found from 134.5 picograms per ml to 290 in the 19 days period. That means they continuously found the significant increase of this troponin I levels. And what happened? And in this myocardial injury, they found kind of biomarkers elevation, and the biomarkers are D-dimer, ferrin, interleukin 6, and lactate dehydrogenase. And by finding these markers, they confirmed this myocardial injury. Coming to this exact mechanism, they don't know, but still, like, there is optional mechanism, direct myocardial involvement mediated by AC2, because AC2 receptor also present in heart and kidney and several tissue types. And murine, but so far there is no studies, but there is one animal model. They found like they're infected with the SARS-CoV, and they found infection in pulmonary vessels, and they found this AC receptor dependent myocardial infarcts in that transfected mice. So that's telling this is the one of the reason, but still their studies are doing about this one. And coming to the arrhythmia is an irregular heart rhythm, which is like slower heart. And they found 16.7% people at arrhythmia and increase from 17 to 45% around in COVID-19 patients in the ICU. And patients, they should monitor with the right side of heart failure, secondary pulmonary hypertension as a result of hypoxia, because hypoxia is the main cause of this lung injury. And coming to the heart failure, newly diagnosed cardiomyopathy, chronic heart failure, they found in the COVID-19 patients. And they did a cohort study with 21 patients with 90 individuals. And they found the development of cardiomyopathy in 21 sick people. And here, this is the case report study, and they taken from the infectious people. And this is a CMR scan picture, cardiovascular magnetic resonance picture. And here, left panel A is the T2 stir, which is like a weight light gradient enhancement of MRI scan. And in this arrow marks indicates that the myocardial is infected, and you can see the epicardium, pericardium, both are like infected. And the B1 is T1W which is like another view. And here also they confirmed the infection, and this left ventricular thickness was like destroyed. That means that's thinned. So that's the reason it has problem to pumping of the blood. It's called a cardiomyopathy. And coming to the drugs, and my previous speaker, Tanya Mahe, and he addressed all of these compounds. Then I removed some slides like while he's talking. And I must keep chloroquine and hydroxychloroquine because this is main compound in my study. And coming to the many antiviral drugs and other drugs are involved, and these are studied in many in vitro and in vivo, even in the patients they're administered with along with this chloroquine and hydroxychloroquine. And they're still finding which one is the best, but they're not finalizing it. So due to the reasons he said, there are some political reasons. Okay, coming to the chloroquine and hydroxychloroquine. Chloroquine and hydroxychloroquine appears to block viral entry into the cells, inhibiting glycosylation of host receptor and proteolytic process and endosomal acidification. These agents also have immune modulator effect through the attenuation of cytokine production and inhibit the autophagy and lysosomal activity in the host cells. So chloroquine inhibits SARS-CoV-2 in vitro with of maximal effect. This is called, it is also called EC50 value. The hydrogen chloroquine EC50 value is 6.114 micromolar. And the chloroquine EC50 is 23.9 micromolar, which means like hydroxychloroquine is much better than chloroquine because it has hydroxyl group and it's doing something which lysosomal infection and blah, 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 something. So there is a main effect of the chemistry. I'm not going to discuss much about because I'm not a chemist. Well, like to go to the next slide. In this, like they tested and hydroxychloroquine in several patients in several with different dosages. So they found the changes of viral effect, and also they quantified many parameters. In the first study, they chose 36 patients, and 20 they given like hydroxychloroquine, and 16 they gave like they left as control group. The reporter they given like 20 milligram by mouth every 8 hours. They compared the control patients to receiving standards, and the clearance at the 6th day they measured nasal swabs, and they quantified 70% in 20. Means they found like 14 out of 20. versus like 12%. That means they found 2 and 16 of the hydroxychloroquine control and respectively. So it is significantly different. The another prospective study, and they choose 13 patients in China, the randomized patient, and they given hydroxychloroquine 400 milligrams. They increase 200. And in between there are some, but I choose like a certain concentrations because I can't give all the clinical studies, which is not useful. So daily they given 5 days plus standard care. Standard care means they give another alternative drugs like antivirals. And the standard care they choose like 1-to-1 fashion. That means alternatively. And they found there is no difference compared with the previous. And the 7 virological clearances similar, like 86.7% versus 93, which is not significant, even though higher. So coming to the another dose, they increased from 400 to 600, and they give like two different concentrations, like 500 and 600. And the 600 they didn't give because it's a high dose. They given like 2 times, like 200 plus 400. Even this, in this case, the patients experienced some illness or some side effects. So these both agents caused the QT prolongation. QT prolongation, I explained in my previous slides, like what is QT, CQRS, and PQRST. Well, and also it causes hypoglycemia and also neuropsychiatric effect or neurotoxicity, and also it's caused retinopathy, it's affected kidney. The baseline electrophysiology to evaluate the prolonged QT is advised to prayer following initiation of the medication because the potential arrhythmias, especially critical ill patients, those who are QT interval prolonging medications such as erythromycin and fluoroquinolones is very bad. They have very high illness with the chloroquine combination of azithromycin. So coming to the explanation about QT prolongation, I just explained about what is the ECG. Here again, I'm telling you, this is the P wave and QRS, and this is the T event. So when atrial starts contracting or breathing, here the initiates the electrical signaling, it reaches to the ventricular, and this QRS complex indicates the ventricular contraction. And when it goes to the T wave, that means ventricular relaxation. And this is the normal ECG. And what happened in QT prolongation? And this QT interval will prolong. And what happened when it's prolonged and there is a gap between beat to beat, and it will destroy the beats and it slows down the heart rate. And that means it is called cardiac dysfunction. It is also called Torsado point syndrome or disease. Okay, Torsado point Torsado point is nothing but the ventricular tachycardia. That means it's slowing heart rate. And that was by QRS complex around the electrical signals, because here it's disturbed because of ventricular and it's prolonged because of the ventricular illness, and it's prolonging the QT signaling or slowing down the T wave. All right, here the Torsado point. Here you can see the clear image of the normal and abnormal or Torsado point ECG signal. And here is P wave, QRS, and T wave, and this is again PQRS. And this between RR, these are called RR signaling or heart rate. That means this is the beat to beat. This is 1 beats, 2 beats, 3 beats, like that. And this is the QT interval. And the panel B, you can see the normal heartbeat. And here the ventricular QRS complex having some problem, and it's slowing heart rate, and it's disturbed the T wave. And finally, it's disturbed another heart rate. And finally, it's missing in between one heart rate, then missing 1 beat, and again see the normal beat. And finally, it's the disturbed heartbeats, but there is no P wave, and it's very disturbed. And the Torsado point disease is a life-threatening arrhythmia and leads to sudden cardiac death. So that's the reason. So high dose of hydroxychloroquine, which leading to the QT interval. And coming to the chloroquine. So I like chloroquine because for the past 3 years, I'm working on chloroquine as a blocker of IKCA channel. IKCA channel, I'm gonna explain you in future slides. So I'm using this small molecule to block atrial fibrillation in a very small concentration manner. So what is atrial fibrillation? Atrial fibrillation is originate the irregular heartbeat at the atria, and it's spread over the heart and completely, finally it's disrupt the complete heartbeat. Here is a normal heartbeat and atrial fibrillation heartbeat. And here is clear PQRS complex and here PQRS. But here you can see the R interval and RR, but in between there's a P is missing. The P means here is a electrical signal is starting at atria and this spreading to the ventricular. So the reason here P is missing, the electrical signal is disturbed at the atria only. You can see in these two videos and the difference between normal and atrial fibrillation. Here is irregular heartbeat started at SE node and is spreading toward the heart. So hope you are understanding this way. All right. Okay, in the action potential, everyone knows about this action potential. In this action potential is a resting membrane potential, and here the sodium channels and rise the action potential peaks, and calcium and potassium ends back to the reversal potential or resting membrane potential. And in this SR means sinus rhythm or normal heart rhythm, and AF means atrial fibrillation. So what happened during the atrial fibrillation, and it's a potassium channel deficiency and it's shortening the action potential. This is the one of the sign or the measurement, electrical measurements. All right, so coming to the potassium channel. So how I'm checking this chloroquine to prevent the atrial fibrillation. So in a basic pathophysiology, acetylcholine activated endorectal potassium current. So in a physiology, acetylcholine is a neurotransmitter. It's released and it's bind to the G protein muscarinic receptor 2. And soon after it's bind, it makes the conformational changes in the intracellular domain. And in the intracellular domain, there are three like GTP alpha, beta, gamma. Soon after it's bind, this beta, alpha, gamma dissociate and binds to the another protein, which is called KR3.13.4, which is tetrameric protein. And this is a major protein can alter the IKCH current. And it makes conformational changes in the intracellular domain, and it opens the channel and all the potassium channels can go out. So then that is the reason it's increasing IKCH current. In chronic atrial fibrillation patients, we see increasing or enhancing IKCH current. So, so far some drugs, but there are no exact drugs to block this IKCH current. So there's one drug called like tercipin Q, which is 21 amino acid peptide isolated from European honeybee venom, which blocks this this channel and it controls going out this potassium current. That means it's controlling the IKCH current or inhibiting the IKCH current. Then we can prevent the atrial fibrillation. But we cannot use this tercipin Q in a clinical because this tercipin Q stays lived in the blood-brain barrier. That means it can also affect on the brain. So we decided to test this one because my PA, Sami, and he's been working on chloroquine several years. And after I got a chance to work with him, I did some patch clamp studies and I found this effect on IKCH current. And this paper I published in 2017 FASB journal. And in this study, we took two different kind of cell lines like HEC293 blank cell lines. And in the HEC293 cells, I transfected with the KR3.13.4 protein, which is exact protein mimic of the IKCH channel. So what happened? I tested the tercipin Q, which is a non-blocker with the green fluorescent at 488. And I did confocal imaging. And what happened here in the blank cells, I didn't transfect here because this is blank and there is no green signal. But here I transfected with KR3.13.4, which is cited in the cell membrane. We can see here clear green color in the cell membrane. And this is clear, this is a merge picture between the cells and membrane. Here, coming to the panel B, I tested both tercipin Q non-IKCH channel blocker and the chloroquine with the patch clamp recording. Patch clamp is a technique which we can measure single ionic current in excitable cells like neuron cells or cardiac cells. So I found IC50 of chloroquine around 1.02 micromolar and tercipin Q around 10 nanomolar, which is already known. So, but this is not like not great like tercipin Q, but still it's inhibiting the channel. It's in small molecular value, like it's 1 micromolar, it's nothing. So we did another study. Like in this study, we tested this chloroquine in humans who are like suffering with chronic atrial fibrillation, and also we did some mathematical modeling. Like mathematical modeling is like bioinformatics way. So one person asked like in the previous session. So we tested these drugs, and we see the blockage of IKCH channel and currents. Here is the case study. the patients, we gave the chloroquine, some concentration, which is very low concentration. So what happened when we gave like 1 or 2 days, it's become normal. And after stopping at 6 days, again, it's showing the AFib. And we started giving at the 10th day. So, and 11th day is going down and 12 days back. And here you can see the clear picture of the normal ECG when we given the drug, and the middle picture is in between the drug, that means at the 11th day. And here is during the atrial fibrillation time. Here you can see the QRS complex and T wave, but there is no P wave in this ECG recording. All right. So these are the mathematical modeling and computational stimulations. So mathematical modeling computational stimulations are like it's a software program. So we code exact the heart by taking pictures, and we give the same coding of the drug, and we do the computational stimulation. So in this stimulation, I don't know, somewhat I couldn't play the video, but this is called electrophysiological rotors, and these rotors is based on this color intensity, and we can see the voltage, like millivoltage or membrane potential, whatever it is. Here in this the 4 pictures in the different time points, we take screenshots. And finally, here we can see this, the rotor is entered here and the transmitter from one place to another place. And finally, it's make green. That means it's terminated the atrial fibrillation at the site of atria. All right, so I checked all the effects, and because my PA also done several experiments and he studied several targeting hypotheses. So there are several aminoequivalent compounds like chloroquine, hydroxychloroquine. There are a few more like primaquine and quinidine. These are like a non-antimalarial drugs. So we decided to check this effect on IKCH current. So the basic technique is like a patch clamp, because with the patch clamp only I can measure the ionic current. So when I measured IKCH current by using automated patch clamp, this is the IC50 value of all the drugs, like chloroquine 1.1 micromolar, hydroxychloroquine 5.4.7, quinidine is something. So I took screenshot or like representative pictures from chloroquine and the primaquine, because these are like a similar structure compounds. I found more blockage with the chloroquine, not with the primaquine. Even you can see the difference of IC50 value. And chloroquine is 1.1 micromolar, primaquine is 47 micromolar. I did same experiment with manual patch clamp, and I found similar values. And this is the comparison between automated and manual patch clamp, and it's almost similar. And recently we decided to write a review, and also we got invitation from a current pharmaceutical journal, and the many other like chemists and many good scientists are collaborating, and we are making this work done. Dr. Brahmachari sir is like a main author in this, and Tangamala and Manoj Kumar, these are like medicinal scientists, and they're helping me with chemical structures. Sangeeta is helping with me chemical composition and structural. And in this study, what we are doing, so chloroquine, hydroxychloroquine, when they given high concentration, like when they increased from 100 to 600, they found a very bad heart problem, like QT prolongation, which is very harmful for the patients. So in this study, we decided, because after chloroquine, testosterone, and malarial effect, there are many compounds came, but they are not completely coming to the market or completely reaching to the people. But when they tested in the laboratory or in vitro or in vivo, they're still seeing more effect than the chloroquine. In their studies, the chloroquine is the control compound, and their compound showing like hundreds or 50 folds better than chloroquine. So, and we decided all the chloroquine compounds are natural products, and it's having aminoequivalent structure. So I chose very few compounds because I'm not interested to put everything, because we're collecting almost 50 compounds, and it's almost done because due date is next week only, we have to submit. Coming to the chloroquine introduction, chloroquine fame is the antimalarial drug, and from the history, which is natural product because it's isolated from cinnamon bark. And in 1820s, Kevin Towe, he's the first isolated, and Perkin, he's tried to synthesize the quinine structure. And first they found the quinine. From quinine, they are synthesized the chloroquine. That's what is called quinoline compounds. Coming to the chloroquine structure, and what is the main importance or structural importance? Here in the quinoline is tetrameric ring, crucial for heme binding. And this is tertiary amine, and this can reduce the basicity, or its main role on the basicity. So this is the importance of structure. And coming to the these compounds, and I choose only 5 compounds along with the chloroquine, and this is the structurally little bit similar, but there are some changes in this side chain or whatever it is. These hybrid compounds have many benefits, including low risk drug interaction and pharmacokinetics to induce a quality based study, and also it can give more effect, or like it can block more powerfully than chloroquine in malarial studies. So in this study, we are proposing this anti-malarial drug could be more powerful than hydroxychloroquine, because these malarial drug are showing more effect than the chloroquine in malarial inhibition. All right. Finally, in the conclusion, anti-malarial drug, so far all the drugs, whatever giving is not so promising, and also they still have to find the drug. So chloroquine and hydroxychloroquine have long-standing history because they have very good effect on malaria, and it's helped many people, and also it's having antiviral effect, and also it's showing a good effect on heart, like by blocking IKC channel, it's controlling atrial fibrillation in the low concentration level. But when it's going to high level, it's going toxicity like QT prolongation and myocardial infarction and arrhythmia and cardiomyopathy, and also at the same time it causes the neuron toxicity like neurotoxicity and renal cardiomyopathy. Renal toxic kindly. So finally, we are concluding, and this hydroxychloroquine compounds in a high dosage is very harmful, but low dosage is useful on malaria, but other aspects, but which is not useful with in antiviral treatment in COVID-19. So in case if you use the similar structural compounds from natural products or whatever the synthesis are more powerful than a non-hydroxychloroquine or chloroquine. So thank you. So I'm happy to address your questions. Please shoot your questions. Hello.
Hello.
Yes.
Thank you for your presentation, Dr. Bujjibabu. And
You're welcome.
I just ask you some questions, like from the participants, we have taken the questions. Is
Sure,
there any possibility to prevent the virus into the cell through receptor by mimicking or blocking the receptor with synthesized molecules?
Synthesized molecules. Okay. I think if the participant listen carefully, the Tangumalai's talk, I think he addressed a little bit about this one. And he targeted many and other receptors, including that ACE2 receptor.
Okay.
Yes.
Another question. Indian corona strain is same with other country strain, or is it different species?
All right. So the species levels. Okay. Well, I read one paper in the Biorca. Even you can find it is free, and everybody can post. They found similar. They did kind of a genetical evaluation, like population genetics. So they found Indian genes have some immune response than other nationals. So that paper could answer these questions. So I think it's yes. I think there is difference. Coming to the strains, I'm not sure about that.
Another question, like any information about how heart feels when corona enters the body?
Pardon?
What is the status or how does the heart reacts when the corona enters the body? Is there any symptoms or anything like?
Okay. So first of all, we can't see or we can't feel the heartbeat. Okay. Of course, the only the person can feel. But patients, like whoever on the in ICU and on the ventilator, so he cannot say like my heart is like pumping or like palpitation. So only thing is the physicians, doctors can find by monitoring their ECG or echocardiography results. That's it.
Thank you very much for your very good presentation and sparing your valuable time and sharing your very good informative knowledge with our participants. And we look forward for in future your support regarding this therapeutic or any regarding this
Definitely.
COVID or any other research. And on this platform, I would like to share with everyone that he's our student from Nani University, and we are proud of you.
Thank you very much.
Bujjibabu.
Thank you very much, madam, for
Thank
this
you.
opportunity. I'm not feeling like I'm presenter. I'm still feeling like I'm your student, you know. And also I'm feeling it's a great
And
honor
he's the
to
person
give a
who
lecture.
was there behind me from the initiation of this webinar. Thank you for your support also. Thank you
Thank you
very
very much,
much.
ma'am. Thank you.
Thank you.
Bye-bye.
Thank you.
Thank you all for your time and patience.
So many likes are there and they're telling it's a very good presentation, Bujibabu.
Oh, that's
Thank
great.
you.
That's great then. Thank you very much.
Thank you. ఎదుగో అవును అలాగే Now I request Dr. T. Padmavati to introduce our next speaker, Gopinath, Dr. P. Gopinath.
Yes, ma'am. I'm here. Can you hear me?
To introduce our speaker, Dr. P. Gopinath, who is Assistant Professor, Department of Chemistry from SRM Institute of Science and Technology, who is going to deliver his topic on molecular biology approaches to target diseases, including COVID-19. So he did his MSc in chemistry from Bhartiyar University, Chennai, PhD in chemistry from IIT Madras, and postdoc from top institutes such as Max Planck Institute, Germany, Technion, Israel, and EPFL, Switzerland. He received many scholastic awards and honors, Best Poster Award from IICT Hyderabad, and he has many peer-reviewed high-impact publications, more than 15, and he has a patent on enzyme inhibitors. He also guided many MSc and PhD students and completed research grants, that is as a principal investigator as well as principal investigator in DST SC SERP, and also now there is an ongoing project that is research grants from DST SERP. Now I invite Dr. Gopinath to deliver his topic. Thank you.
Thank you, ma'am, for your very kind introduction. Can you hear me? Hello?
Yes, we are able to hear.
Yeah. Thank you very much, ma'am, for your kind introduction. And I'm sharing my screen. So I take this opportunity to thank the organizers for inviting me to this international webinar series. And I'm thankful specifically to Vijaya ma'am for that special invitation. Like many other, like two speakers spoke before me, and they gave a very, very detailed introduction about what is COVID-19, and I'm sure all of the participants are aware of the current situation going around us. So I don't want to take much time in introducing many things related to COVID-19 initially. But what I wanted to do in this 30 minutes is we have learned and developed so many biological approaches to target several diseases, and the lessons learned from these molecular biology approaches, whether we can expand it to combat the COVID-19. So this is what I'm trying to convince you over 30 minutes. And before getting into my research topic, let me quickly remind some terminologies related to this, what I'm going to talk about. That is a chemical biology or the molecular biology. So chemical biology is something like understanding the fundamental mechanisms of various biological process at molecular level. Okay. Whereas a molecular biology is something like a branch of biology that deals with the structure and the function of macromolecules. Macromolecules here, I mean proteins and nucleic acids. Since my topic is only related to the proteins, I hope you are seeing my cursor as well. So I will be focusing only on the protein context. So proteins and macromolecules, as you know, they are formed by amino acids. So I arranged my talk into 4 different aspects. So molecular biology approaches 4 different sections. 1, Alzheimer disease, 2 Huntington disease, 3 cancer, and then I will come to the COVID part at the end of my talk. So I took one protein, one disease approach. For example, tau is a protein associated with the Alzheimer disease. HTT is a Huntington protein that is associated with the Huntington disease. USP2 coming under the class of D ubiquitinases enzymes comes under the cancer. So if you target, in simple, if I target tau, I'm going to bring some therapeutic outcome for Alzheimer disease. Okay, if I target Huntington, I'm going to bring some kind of therapeutic target of the Huntington disease. So if I'm going to target USP2, I will be able to bring some therapeutic outcome for cancer. So what are the possible or what are the drug targets associated with the COVID-19 is what I will be touching at the end of my talk. Since I'm going to talk about target proteins, what are target proteins? So we have several proteins in our body, hundreds and hundreds and hundreds of proteins. Okay, so not all the proteins can be targeted. For example, a billiards game, that's an analogy I would like to show. So you're going to hit a ball, and that is going to create a cascade of events. So that is the first ball which we are going to hit is called the target protein. So we need to find a judicious way to figure out the target protein. So this is more important because this is going to be the key important factor which will eventually end up with the success rate of combating the disease whatever you are targeting. Since I'm talking about proteins and proteins, let's understand how these proteins are being constructed and destructed in the body in the bigger context, what we call as a central dogma of life in the context of proteins. Okay, so construction is something like you're creating the protein. So how the proteins are created, we have DNA. There is a transcription process where you get the RNA copy, and then the RNA translates the genetic code into the protein. So once a protein was synthesized, this undergoes several post-translational modification. I want to emphasize this word because this is what I will be keep using in many throughout my talk. Post-translation modification is nothing but a modification, any chemical modification, for example, acetylation, nitration, phosphorylations. So what is that post-translational modification? Is nothing but a modification on the protein. So translation, so you are doing some modification after the translation, that is post-translation. So these are the all possible modifications you can do. So we in general call it as PTMs, post-translational modifications. Okay, so in 2 of the proteins I will be emphasizing on the phosphorylation stuff, and in one case I will be focusing on the ubiquitination stuff. So once these proteins are formed, these PTMs are important to translocate from one part of the cell to the another part of cell. It does its function. So once its function is done, the body decides to eliminate the protein. So that is the destruction process. So okay, the protein was made, it is done with this function. So it can be eliminated in 2 different mechanisms, autophagy or ubiquitin proteome system. Okay, this is known as UPS. Okay, and how does it happen? If our body decides to eliminate this particular protein, this will put a ubiquitin tag on the protein. So once it puts a ubiquitin, this is a signal that this is a protein which is undergoing the degradation. So this will undergo, okay, the protein will underwent ubiquitination, and this ubiquitinated substrate will undergo to, it will be directed to 26S4 particle, and here the protein will be degraded. So this is about a general proteins, how it is being formed and how it is being destructed in our body. So now I will introduce first protein. Before that, so let me go back, and I will introduce this Alzheimer's disease and Huntington disease. This comes under the category of neurodegenerative diseases. Okay, so I will give a brief introduction about the neurodegenerative disease initially, and then I will move on to one protein, one disease as a key in the presentation. So neurodegenerative is anything which comes under the umbrella where it affects the primary. So anything which affects the neurons in the human brains that leads to the disease, and this comes under the category of neurodegenerative disease. So what happens in Alzheimer's disease or in Huntington disease, there are some proteins as targets, but I am focusing on tau and the Huntington. So in the normal, the tau is doing its function, but in the diseased situation, the abnormal function undergoes misfolding and aggregation. Aggregation in simpler terms is nothing but a precipitation. So once you have a native monomer, there are several reasons why it is getting aggregated. Okay, so there are several researches done in this line, but that is a big ocean and I'm not going to touch upon that. So trust me, now I have a monomer and it's because of some misfolding. There is oligomerization and then it forms a fibrillar like amyloid structures and then it gets precipitated or aggregated. So this leads to the neurodegenerative diseases. So there is, until now, there is no cure for Alzheimer's and Huntington disease. And so there are like 40 to 50 years people are working in this area, but several drugs failed in the clinical trials, mainly because of the poor understanding of the pathology. This is due to the lack of tools and more accurate models to understand and target this particular disease. Our approach is to target this tau. So we utilized a total chemical synthetic approach. So this is a schematic representation of how we executed this project. So this is a tau protein where the length of this protein is 441 amino acids. So like I said, PTMs, post-translational modifications, these proteins can undergo a plethora of wide variety of post-translation modifications starting from phosphorylation, acetylation, ubiquitination, glycosylation to glycation and sumolation and so on. What we are interested in this particular protein is a K18 domain. This is also a protein. And we wanted to understand what happens if we put phosphorylations at 3 different sites, that is S258, S262 and S356. So why is it important in the Alzheimer's patients? When you take the brain and they look at the postmortem brain, they see that these phosphorylated sites are present in the Alzheimer's patients. Until now, it is not clear whether this phosphorylation leads to the aggregation of this protein. Okay, so that is the reason why, that is the reason we wanted to systematically synthesize these proteins and wanted to understand its aggregation behavior, and moreover the crosstalk between one phosphorylation to the other. So end of the day, the proposal is very simple. I have a tau protein. What are the factors which will affect its aggregation? If I am stopping the aggregation, that means I am going to bring a cure for the Alzheimer's disease. I want the whole idea is this protein should not get precipitated or aggregated in the brain. Okay, so this is what we have done, and then we did an aggregation in cellular seedings and microtubule assays that I will come up in the next slides. So the first part is like the total chemical synthesis. So this is 130 amino acid containing protein. We broke this protein into 3 fragments. Okay, the green block, the red block, and the orange block. So in order to put these pieces together, we use a technology called a native chemical ligation technology. So there are 2 conditions where this NCL will work. For example, the C terminal of a peptide fragment should contain a thioester moiety, and then the N terminal of the other fragment should be a cysteine. If this is not the case, the reaction will not happen. Okay, so we made this proteins, so these peptide fragments through solid phase peptide synthesis. This slide looks very simple, but it took nearly 1.5 years to prepare all these proteins in a site-specific manner. So we made this building block in the orange and then in the red, and then we ligated this together. So we got this particular fragment 4. So here we wanted to make 2 fragments, one without phosphorylation. One without phosphorylation and one with phosphorylation, because we also want to have the control of proteins with and without phosphorylation sites, right? So we have 2 fragments from this orange block. Since this doesn't have any modification, we have to build only this red, and in this green we made 3 different variants, one without any phosphorylated sites, one with 1 phosphorylation site, and another variant with 2 phosphorylation sites. So when we have all these pieces together, we stitch these pieces using native chemical ligation. So you do this, this reactor, and you see the cysteine is in the protected form. You deprotect it so that you have an N-terminal cysteine, which is prerequisite for an native chemical ligation. So this cysteine reacts with this thioester of the green piece, and then you are able to get this particular protein. So we made 4 different proteins. The first protein doesn't have any phosphorylation. The second, for example, here, K18 wild type, it doesn't have any phosphorylation. K18, this variant have 1 phosphorylation site at 356. this protein have two phosphorylation sites at PS 262 and PS 356, whereas this protein has phosphorylation on all the three expected sites. So once we have these four proteins in hand, we went ahead and to see how these proteins aggregate. I'm not going into the technical details. What we got, or what is the take-home message here is, when I have all the three phosphorylations on the K18 protein, the aggregation is inhibited. So far in the literature, so many, even in the textbook chemistry, people argue that phosphorylation will lead to aggregation, but our studies clearly tells that phosphorylation inhibits aggregation, which is a really important lesson we have learned from this particular study. And moreover, we also did the seeding ability in the biosensor cells. This was done in collaboration with Mark Diamond from US. So when we have an K18 protein in the presence of heparin, this will form the aggregation. So if it is doing any seed destruction, so this is piling up. So it's aggregating to on one top of the other. And because of this biosensor cells developed by Mark Diamond group, you will see a fret, that is an emission from this particular cells if this is undergoing aggregation. But if it is not undergoing aggregation, you will not see this particular fluorescence. Okay, so this is based on the fluorescence and based on the FACT studies, they quantified it, and whatever we found in the aggregation studies, that triphosphorylated K18 inhibits aggregation. This was further corroborated from the Diamond's studies. So this is a take-home message from this particular project, that we made a K18 protein with these fragments, and then we assembled, and then we studied its aggregation, and it's a cellular process. The reason why I am mentioning this molecular approaches, so everywhere now the protein is being targeted. So if you wanted to understand the protein, we need to make the protein. So in order to make the protein with the very specific modifications of our interest, you should develop molecular biology approaches. So this is one such approach. And the second one is related to the Huntington disease I mentioned. So briefly, the Huntington disease, this is a normal brain, and this is a brain from the affected people. So in the normal people, we will have a CAG coded for glutamine. Okay. So in the healthy neuron, there is less than 36 CAG units, meaning that I have a polyQ until 30 less than 36. But in the case of the patients, you will have more than 36 CAG units. So whenever there is a mutation, this is genetic disorder. So whenever there is a more CAG units, that means they are diagnosed as Huntington patients. So you look at the size of the protein, it's 3,142 amino acids. It's such a very big, huge protein, and we don't have the tools to handle this big proteins as of now. So, but what we have found in the patients, for example, is that the N-terminal fragments, okay, the N-terminal fragments of this particular big proteins are neurotoxic, and they are really important for the pathology. So we wanted to understand the post-translation modification in the fate of this particular protein in the context of Huntington disease. So this big protein, we are focusing on Huntington 1 to 171. Okay, so usually the first amino acid is methionine, which is processed. So we always term it as 2 to 171. So what we are interested in is what would be the fate of this particular protein when we have phosphorylation at T107, that is threonine 107, serine 116, and serine 120. So if you want to know what is the fate of this particular modifications, we need to make the proteins. So what we envisaged here is a two-fragment approach. In the last project, we used a chemical protein synthesis approach, and one can ask, so, okay, why don't you make all the 171 amino acids synthetically? That is not possible because of this polyQ. Because in the Huntington patients, there are more than 43 polyglutamines, which is not possible to make synthetically. So what we have to do, we have to seek the help of nature, that is bacteria, which has the ability to synthesize this particular proteins. So we take from the bacteria this protein, and then we will try to switch. What we call it as a semi-synthetic approach. So part of the comes from the nature and part comes from the synthetic laboratory. So here is the semi-synthetic scheme. So we are interested in 2 to 171 protein, and then we made into two fragments, that is 104 and once. There are several rationales why we are working on two fragment approach. That is out of the scope of this particular topic. what I'm trying to convince is the approaches, how we are able to make the proteins, and what are all the required studies we can make. We need to make the proteins. That is the only point I'm trying to highlight throughout the topic, throughout the discussion. So here we have 2 to 104 fragment. This was recombinantly expressed. I have one slide after this and a flowchart how this works. So this is, you can make 2 to 104 protein, and then as I told you, we need a C-terminal thioester to do the native chemical ligation. This was installed using an in-time approach. I'm not going to the technical details, but so this is not an easy task to install a simple functional groups using bacteria, using the recombinant protein expression. Okay, so the other piece of this 2 to 171 is like 105 to 171 protein fragment. This was also expressed using bacteria. So we have the SUMO tag, which was cleaved by a ubiquitin-like protease. So this is a very specific protease which cleaves this particular linker, and we obtained these 2 fragments, and then we stitched this together to get the native Huntington 2 to 10171 with 23q. So when I say 23q, this is present in the normal people. When I say it's 43q, it is the mutant version that is present in the patients. So we have made the native one here, but our goal is not to have this native one. We wanted the site-specific phosphorylation sites installed at 107, 116, and 120. So this is a proof of concept that we can make proteins without modifications. So these are the data which clearly analyze the formation. So I'm not going into the details of how we have characterized these things. So this is the flowchart of how the protein is being expressed. So we have a plasmid, the recombinant plasmid. So you encode the protein sequence which you want to really express, the overexpression model. So you do a transformation to the bacteria, the pre-culture and the big culture, let's say in a liter to 12 liters scale. Once you have this bacteria, this can be centrifuged to get the bacterial pellet. And this bacterial pellet, so you now the bacteria synthesized your protein. Now you have to take this protein out from the bacteria. So first you do a cell lysis. So that means the bacteria will be, the bacterial cell wall will be smashed, and then the proteins will be in the solution. So the bacterial cell plate, so whatever you get from this lysis, you will be doing an affinity purification using FPLC. The FPLC, so it's fast protein liquid chromatography. So there are some techniques why we are doing this and so on. I'm not getting into this, but trust me, this is really a very nice technique in order to access the protein with your interest. So you do an FPLC with the ULP cleavage, you will get your protein of interest. But if you want to do a thioester, as I said, the C-terminal peptide fragment should be a thioester. If you want to really do a thioester, you need to do a splicing with a Mesna unit. Okay, so by doing this one, I got 2 different protein fragments. So what is still missing is the methodology to introduce the phosphorylation in this particular piece, the second one. So here there are 2 approaches. Like I have this fragment, I can do an in vitro kinase. That means kinase are enzymes which phosphorylates the residues like serine and threonine. But in this particular case, we can also look for the chemical protein synthesis. But when we attempted to do this chemical protein synthesis, it's really hard to get this because there are so many aggregation domains, and the proteins are all getting precipitated, and we are not able to continue the synthesis. So we went to this, we took the second fragment of this protein, we reacted with kinases, and then we obtained a very site-specific phosphorylation. We did extensive characterization of these proteins. I'm not going to touch on those details, but this Huntington 105 to 171, we expressed it through bacteria, and then we did an in vitro kinase screening. We utilized more than 83 kinases. That means each kinase again is a protein. So we did kinase screening for 83 kinases. That's a very big screening we have done, and we come up with a very site-selective kinases at T107, S116, and both 107 and 116. Even though 120 is of our interest, we were not able to find a selective kinase to do it. But in the Huntington patients, only S116 is of most important. That is the PTM present in the Huntington patients. We also developed 2 antibodies which would recognize T107 and PSS, the phosphorylated serine and threonate 107 and 116. So with all this, again the native chemical ligation, but this time we have the phosphorylated, that is a site-specific phosphorylated one in the fragment itself. So this allowed us to make Huntington 2 to 107. That is not only on 23q, that is in the normal humans, that also in 43q, that is in the Huntington patients. So with 23q and 43 variants, and along with mono and diphosphorylations, we made 8 different proteins as of now, and currently we are investigating how these proteins are or how these post-translation modifications are important in the diseased situation. So these are some examples from our earlier studies, how to make proteins, how to make site-specific PTM modified proteins, and how to study its behavior, and how to use that as a particular target in the disease. So this is the second part of the. I told that I have discussed. I have made this presentation the 4 parts. One with the tau, that is Alzheimer, which I discussed already. The second is Huntington, which I just finished, and the third one is related to cancer, that is deubiquitinases class of enzymes. So before getting into deubiquitinases, I should introduce what is ubiquitination first. So ubiquitination, it's a 76 amino acid containing protein. So I already have a protein, so I am attaching a small protein as a modifier on a bigger protein. So this is already a bigger modification. And also in the very first slides, I told that if the system decides to eliminate the protein, ubiquitination is the key labeling our nature is utilizing. So ubiquitination is a very, very important PTM modifier. And as I forgot to mention that every post-translation modification, there is another set of enzymes which will reverse a post-translation modification. For example, on a protein, I'm putting a ubiquitin on top of it, and there is another set of enzyme which will remove this ubiquitin out. Okay, that is what I am calling it as deubiquitinases. So this particular ubiquitination itself is a very complex machinery. I'm going to tell with this slide. For example, I have a protein, and ubiquitin can sit on this particular protein. So there are 8 different ways where this can be anchored. For example, I have lysine, K6, K11, K27, K33, K48, K63, and the C terminal. So there are 8 possible ways a protein will get modified by ubiquitin. Okay, and assume that I am putting 2 different ubiquitins on a substrate, 2 times 8. And assume that this ubiquitin chain is being elongated. This is a polyubiquitinated chain. And this ubiquitin can make a polymer kind of thing. So ubiquitin, 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 ubiquitin. Now I told in one ubiquitin you have 8 possible anchoring sites. So dipeptide, ubiquitin, ubiquitin diprotein will have another 8 possibilities. So here another 8 possibilities, here another 8 possibilities, and then here another 8 possibilities. So the reason why I am emphasizing this particular machine is very, very complex, that depending just on the linkage, it will decide the fate of the protein. For example, here simple monoubiquitination protein, monoubiquitin, okay, this will be sent to the DNA repair. I have a protein with 2 monoubiquitins, it will be sent to the endocytosis or proteasome degradation. So when I have a polyubiquitins, it can go for cellular signal, kinase activation, or DNA repair or endocytosis. Just based on the ubiquitin labeling, it is going to dictate the fate of the protein. So that is the reason why ubiquitin post-translation modifications are very, very complex and important for the normal functioning of the body. So now it is the reverse of deubiquitination, the ubiquitination, that is the deubiquitinases. So the substrate protein can undergo ubiquitination, as I said. This machinery itself again complex with E1, E2, and E3 enzymes, where we have thousands and thousands of E1, and then several hundreds and several few of E3 enzymes. So even just to put one ubiquitin on the protein, the enzymes have to work together in the presence of ATP. So after having one ubiquitin, we can undergo editing process. Again, even with a cascade of enzymes to put polyubiquitins, and then this X represents the chain or the linkage what I have discussed about. And all this leads to a very complex machinery, and this can be reversed by a set of deubiquitins. Let's say I have 4 ubiquitins on it, the DUBS acts on it, and then it can bring it to 1 ubiquitin. or this one ubiquitin can be chopped off and bring it back to the normal protein. So what is the need of this particular DAPs? So this particular DAPs, what I'm talking about ubiquitinases, comes and can be classified into 5 different categories based on the mechanism of action. I'm coming directly to the importance of these DAPs. These are protein targets overexpressed in various pathologies, including cancer, neurodegenerative disease, and viral infections, which is the current context of our discussion. So what we have done is a USP2 test. So we are targeting USP2, that is nothing but a deubiquitinases. So when I target USP2, I'm going to target the prostate cancer. So in order to do this, we need to develop an assay. So this is what we have done precisely. So as I said, deubiquitinases will chop the ubiquitin. So ubiquitin is needed. So the substrate itself contains 80 to 100 containing protein. It's a big challenge to make it. This itself is a big work in tax in order to make this particular protein. So you have 82 amino acids properly positioned thread pair. So what happens if I wanted to target this DAP, that is nothing but ubiquitin specific protease 2 in this particular case? If the DAP is active, this DAP will go and clear this amide bond. So this one piece will become 2 piece here. So you will see a substrate that is fluorescence. If it is together, if you shine light, that is, so I have a blue color here MCA and DNP is the quencher. So when I irradiate MCA, the blue color, whatever the emission comes from here, it will be absorbed by this thread. So you will not get any fluorescence if it is together. But if the deubiquitinases clears this particular white bond, then you will see the fluorescence. So when I am putting an inhibitor, I inhibit, so I will not see any fluorescence here. So if it is not inhibiting, that the enzyme is very active, that means it will cleave and then I will see a lot of fluorescence. So based on the fluorescence readout, I will be able to understand the activity of this particular target. So we did a lot of studies in the small molecular inhibitor, that is ROS. ROS is an inhibitor. So this is a beta currently in the clinical trials for several cancers, including solid tumors. The mechanism of action of this particular molecule is it will create a reactive oxygen species when it comes in our body. So this reactive oxygen species, what it will do? It will oxidize the particular protein. So USP2 comes under the category of cysteine proteases, wherein the cysteine is in the catalytic site, and that is responsible for its catalytic activity. So when you produce a ROS, it is going to do an oxidation of this SH to sulfonic, and sulfinic acid, thereby inhibiting the enzyme. So this is the mechanism of action of this small molecule. So we developed an assay. We are targeting this particular DAPs in order to combat prostate cancer. So several studies were done. So this particular assay development, this chemical synthesis of protein itself is a big challenging task, and this is a strategy we followed. This is mostly a solid phase peptide synthesis. So we need 2 fragments, that is ubiquitin 1 to 45 thioester, and then the cysteine on the N terminal of this particular peptide fragment. We installed DNP and MCA in this fragment itself. So this was coming from this particular resin synthesis. So this is solid phase peptide synthesis. So we got these 2 fragments, then we ligated it, and once we expose this particular substrate to the DAPs, it will cleave, and then you will see a fluorescent readout. So we have done quite extensive SAR studies with small molecules, especially we are interested in quinone-based molecules. What we have done, to cut the long story short, we very systematically studied ortho and ortho and para quinones, and what we found is only orthoquinones. This is directly an enzyme assay against USP2. And all the orthoquinones are showing excellent activity, whereas a corresponding paraquinone is not at all showing any activity. We wanted to address why. In order to do this, we tried to understand the electrochemical potential of this particular beta-lactone, and it's a para analog. But surprisingly, we found that both are equal. But still the puzzle is not solved, because theoretically, if EF is going to be same for ortho and para isomers, it should behave the same, but only the ortho compound is more active than the para compound. So we did quite extensive cyclic voltammetric studies. I'm not going to much details, but one particular point I would like to emphasize here. So this is ortho beta lepacone, and this is a para counterpart of this one. So when we expose this into the electrochemical catalysis studies, you see a very sharp red color catalysis coming out from only from the beta lepacone, not from the alpha lepacone. So we propose an 1 electron, 1 proton transfer of this particular molecule, which is interacting with oxygen. For example, I'm going to generate the reactive oxygen species. So the oxygen, it is very difficult to reduce in our bodies, So we need an electrocatalyst to bring the EF of this particular oxygen. So this particular molecule is acting as an electrocatalyst, which brings the EF of the oxygen from minus 0.53 to minus 0.24. It might look technically difficult to understand, but what I'm trying to take the message here is we are able to distinguish ortho from para when we expose it to the electrochemical studies. That's the take-home message from this slide. So we did quite extensive SAR studies with so many modifications on the molecules. We did an in vitro screening against USP2. We got this IC50 values. We correlated that with an electrochemical electrocatalysis. Both are going hand in hand, and very good correlation of EF with the inhibition profile of USP2. And finally, we corroborated that with the cellular studies doing apoptosis with the DU145 prostate cancer cell lines, where this particular USP2 target is overexpressed. So this is the third, like we continued this targeting USP2. In another study, we screened more than 90,000 compounds in a high throughput format. So we have used the same quenching pair assay. Remember, this is a protein which took 4 to 6 months for us to prepare only this substrate. And based on, after we have this one, this is a commercially available compounds. We screened more than 90,000 compounds. We ended up with some kind of paraquinone interesting scaffolds. And then again, we did a quite extensive SAR studies to understand why this particular para is important. So in this paraquinones, we noticed a very specific pattern of NCONH. So any paraquinones which have this specific particular arrangement of atoms is going to be again a very excellent Ross generator. The Ross generator meaning that it is going to create more and more hydrogen peroxide, which will oxidize the catalytic cysteine of your target protein. So the experiments was further quantified with the electrocatalysis, and the amount of hydrogen peroxide was further quantified using a chemiluminescence assay, which was done in Doron Shabaz's lab in Israel. So, and we also tried to make a peptide inhibitors in this particular case. So in this third case, what I'm trying to understand is you need to have an enzymatic assay if you want to particularly target a protein. So one protein where we are correlating with one disease. So it means that we need to have a robust tools to target this particular protein. So that is what we have done in this particular case. It is USP2. So coming to this, so this is a quite summary of this third part of my talk, that we have come up with so much of small molecules that is targeting USP2, that is a deubiquitinase enzyme that is overexpressed in prostate cancer cells. So whether now the question is, okay, we have learned so much about the molecular biology approaches to target, let's say neurodegenerative disease, Alzheimer's, Huntington, and USP2, for example, in the case of cancer. Can we extrapolate this to the current scenario of COVID-19? So since already the speakers gave extensive introduction about the coronavirus, I would like to take only one point to emphasize here, that we have 7 coronaviruses here, and we are now talking about SARS-CoV-2. What is the difference between the SARS-CoV-2 and these two other coronaviruses about the symptoms? In the case of MERS and SARS, we were able to see the symptoms in 2 to 3 days, and we are people were able to see blisters, and that is the reason why they were able to contain the disease, not spreading across the world, but only few countries. But SARS-CoV-2 is quite dangerous. The main reason, to my opinion, is we are not able to distinguish between the people because it is mostly asymptomatic. Sorry, is it time up? Okay, so this is a model most of us heard today. So what we have proposed here is to targets the one on ACE2. So ACE2 is a receptor which invites this particular virus into our body, and this TMPRSS2 is priming this. If you are able to inhibit this TMPRSS2, if you're able to this CL-Pro, that is nothing but main protease, we are able to combat this particular COVID-19. So people have done extensive research was already started by two people, Rolf Hingland from Germany and Zingman from China. So they developed molecular biology tools to understand this particular protein, and they have already produced these proteins in good quantity. And so what we are, in my opinion, we need to adopt or find a better way to produce these particular proteins and develop an enzymatic based assay. Then only we will be able to find out several inhibitors. So this is a crystal structure by these two groups, and they have come up with two different inhibitors. And the most beautiful thing about this particular two inhibitors are having the same warhead. Only thing is we have a beta amide inhibitors. Here we have a Michael acceptor, but just the backbone and the interacting moieties are same. So with this thought, I'm concluding my talk that we can extend our molecular biology approach to target main protease and TMPRSS2, so that we can develop potential inhibitors in a very quick manner, and especially in the absence of BSL-3 facilities in our country. So I thank all my supervisors for their constant support and encouragement, and I thank you all for your kind attention.
Thank you, sir, for your good presentation. And
the floor is open.
You have two questions here.
Yeah, please.
I read in the news that antiprotozoal medicine, ivermectin, followed by doxycycline for 5 days.
Okay.
Worked on COVID-19 patients in one country.
Okay.
Not in India, outside the country. Is it true? Please enlighten.
I haven't come across this news, but my opinion is these drugs are curing the symptoms. Okay. But if this is going to be a cure for COVID-19, it is going to be a really big news. So what I understood from this particular drugs, they are going to treat the symptoms that probably might subside. But probably I feel that there should be more studies across these lines.
Another question, sir.
Yes.
Chloroquine and HCQ has side effects.
Yes.
Like it affects heart. Then how can we use it as a drug for COVID-19?
So this is about a life and death. See, if I'm about to die, I don't care about the side effects, toxicity, because I'm going to die tomorrow. If something is toxic, if it is able to, prolong my life for another 5 years, am I going to say no? It's about the balance between what you're undergoing. See, whenever the clinical trials is only for the critical patients is what they are trying, not for the one who having a very less symptoms. So for critical patients, I don't think a small side effects is going to be a big deal, because if you're not treating them, they're going to die tomorrow. So in that context, it is important to balance these clinical trials for two different categories: one with severe disease and one with very light symptoms.
Thank you, sir, for your presentation, and for sparing your valuable time and interacting with the participants. Thank you.
Thank you very much, ma'am.
Thank you, sir. Our next speaker is Dr. Sivakumar, and I now request my colleague, Dr. T. Padmavati, to introduce the next speaker.
Thank you very much, ma'am. It's indeed my pleasure in inviting Dr. P. Shivakumar, Associate Professor, College of Pharmacy, University of Pharmacy.
Dr. Gopinath, sir, please stop sharing your screen. I request Dr. Gopinath, sir, to stop sharing the screen. Sir?
So I'm sorry about that interruption, but it's indeed my pleasure in introducing Dr. P. Shiv Kumar Garu, Associate Professor, College of Pharmacy, University of Pharmacy, University of South Florida, Tampa. He's right now in South Florida, and he's an alumni of Andhra University. He did his PhD in the prestigious institute, Indian Agricultural Research Institute, where the thesis he submitted in the C.H. Singh University, formerly the Meerut University. He's a pioneer in hyperoxia-induced cardiac remodeling research, and he published more than 35 peer-reviewed high-impact research articles. His total citations till today are 1152 and authors H index 17, I-10 index 23. He's now currently holding multiple NIH grants. He's course coordinator for translational pharmacogenomics course for the third year Pharma D students. He's a reviewer of many international high impact journals and reviewer of many funding agencies in USA like NIH, AHA, Merck, DOD, and worldwide UK, NOW, Netherlands. He's advising many PharmD master and PhD students at University of South Florida, Tampa. He has many research collaborations with Indian scientists to the tune of 4,30,649 US dollars for a period of 3 years, for 1,49,500 US dollars for 2 years, and $25,000 for studying COVID-19 for a period of 1 year. And there are two projects that are pending for sanction, each for 2 years, and the grant for 295,000 and 149,000 for COVID-19 and Alzheimer's studies respectively. I now invite Dr. P. Shiv Kumar to present his talk on effect of hyperoxia on cardiac remodeling in ICU under mechanical ventilation, a potential mortality factor in COVID-19 patients. I thank the organizers and the speaker for the opportunity given. It's over to you, sir.
Thank you very much. Hi, everyone. I hope everyone is doing good and safe. It's very unfortunate to meet you through virtual conference or virtual meeting. But at the same time, I congratulate our organizing committee to take these unforeseen conditions into an opportunity and make a webinar. I don't know if we have a physical meeting, in-person meeting, I don't know how much turnout would be. It may not be definitely this much turnout. So this is a good opportunity. So it's a good example of how to convert an unforeseen situation into an opportunity. So I congratulate again the organizing committee. Okay, so the talk I'm going to give is mostly about the cardiovascular conditions in the COVID patients. So what I'm going to talk is I'll try to convince you that the mechanical ventilation is the one that causes mortality rather than the coronavirus infection. Let me see how much I can convince you. So going forward, so I want to give more emphasis on cardiovascular results than the COVID. So I'm not going to repeat the same introduction about the COVID, which previous speakers already would have given enough introduction. So, but I'll give you very brief. So what is SARS-CoV-2? So SARS-CoV-2, I think most of you know that it's a severe acute respiratory syndrome coronavirus. So previously there was one coronavirus, which we had epidemic, but it's not severe like this. This is a second coronavirus, which is first detected in Wuhan, China, which is commonly called COVID. COVID is a coronavirus disease, COVID-19. So right now, as of now today, there are more than 6 million infected patients with close to 400,000 deaths worldwide. Definitely, United States stand first in the list in total number of infected patients, and India is running towards the top list, top 5. No doubt, with a proper full diagnosis, India would beat US. So we hope it's not going to be true. We hope that our Indian government take a proper steps and reduce the cases. So what is the association of coronavirus with the cardiovascular disease? From the initial reports from Wuhan, from the clinical data, so there is a publication, JAMA and other medical journals clearly suggest that 27.8% of the patients infected and hospitalized due to coronavirus showed cardiovascular injury and cardiac dysfunction. So which is a clear indication that 27.8 is really a major chunk, definitely a considerable number of patients, statistically significant. So among these patients, the common cardiovascular symptoms observed are elevated levels of cardiac troponin. Troponin is a serum cardiac marker. So just like you go to annual physical or blood tests, when they check your blood test, they also, if you are above 50 or 60 years, or if your electrophysiology or ECG recordings are not good, what a physician do is they will check for cardiac troponin in the serum. So that's a good marker for cardiac injury. So when the cardiac cells die, these troponins release a lot in the serum. So they're similar to lactase dehydrogenase, LDH. LDH also is a good marker for cardiac injury. So these patients are found to have high levels of serum cardiac troponin, and arrhythmias, which is evident by ECG recordings, and also cardiac dysfunctions. Cardiac dysfunction is a functional abnormality of the heart. I will tell you more in the next slide what is cardiac dysfunction and how to determine the cardiac dysfunction. And myocardial injury. Myocardial injury is a kind of, let's say in a layman terms, we can say that cell death. So if the myocardial or cardiomyocytes, whenever I say it means that the cardiac cells, it's a simple cells of heart. So how the heart is made of, those cells are called cardiomyocytes. So when the cardiomyocytes die, then that causes the injury. So that's called myocardial injury. So these are the very common symptoms found in COVID-19 infected patients in Wuhan. Later on, if you check in PubMed, there are a couple of other publications from Italy and other countries shows that cardiovascular symptoms are found in COVID-19 patients very often. So let's go into the cardiovascular disease. So I would like to take a little bit more time on explaining cardiovascular disease and their terminology, what are the pathophysiology, so that when I go to my research results, you can understand. So without a proper introduction, I don't think you will understand. It will be like peaks and some diagrams. So let me give some emphasis on what is cardiovascular disease. So we know that according to the World Health Organization, WHO and the CDC, cardiovascular disease or cardiac deaths are number one cause in worldwide, and of course in US. So according to the data, around 17.9 million deaths each year is recorded. So which is about 31% of the total deaths, which is a major chunk. Definitely, it's a big problem. So the common pathophysiological cardiac pathophysiologies includes very commonly found pathophysiologies are left ventricular hypertrophy. So you know that heart have left and right ventricle. Let me show you. Okay. So if you see this diagram, this is a normal heart and this is a hypertrophic heart, right? So if you see that the heart have right and left ventricles. So the left ventricle, for example, always the takes over, I would say, more than half, maybe close to 3/4 of the entire heart is left ventricle. That's a big chunk of the heart. So right ventricle is small and atrias are obviously very small. So left ventricle is the major chunk of the heart that takes over the space. So left ventricular hypertrophy is a very common pathophysiology. So what is hypertrophy? So I would give you a little bit introduction of hypertrophy. So if you see this diagram, these are the ventricles, left and right ventricles. Each ventricle have a lumen or a cavity where it holds the blood. And when the heart contracts, the blood is pumped out. When it expands, the blood takes in. So it takes in the blood and pumps. So that it works as a motor pump where it circulates or the pump the blood into your over the roof tank. So heart is like a pump, motor pump that pumps the blood to all rest of the body throughout the organs. So the ventricle or space is a major space that pumps the blood. So when the heart contracts and expands, ventricles contracts and expands in due course of pumping the blood. So the mechanism is that the cardiomyocytes or the cardiac cells have a contractile function. So this is called contractile function, like a rubber or plastic. That's called plasticity, and they have a very nice flexibility and contractility. So when these cells lose the contractility, then the pumping ability of the heart decreases. So obviously, even the heart beats normally, the pumping, the percentage of blood that pumps goes down. So that is called the next problem, diastolic and systolic left ventricular dysfunction. So each one is interrelated. So the pumping ability of the heart or cardiomyocytes are the plasticity or the contractility of the heart cells, it decreases because of left ventricular hypertrophy, which eventually cause diastolic and systolic dysfunction, which eventually pump less amount of blood. Even though they beat faster, the amount of blood that pumps will be lesser. So the reason is, if you see these two hearts, the hypertrophic heart, if you see the heart section, longitudinal section, the thickness of the wall is increased significantly. So, but here it is normal. So you may have a question that how the thickness of the wall increases. So is it because of number of cells increases, or the size of the cardiomyocyte increases, or something else is happening? So to go to the basic knowledge of cardiomyocytes, so these are post-mitotic cells. Cardiomyocytes are post-mitotic cell, meaning that the number of cardiomyocytes in a heart in a particular individual is same until the death. So they don't divide. So they will be same. So the number of cells will be same. So how the heart thickness is increasing? So there are two possibilities. One is the cardiomyocyte cell size increases, or something else is accumulating between the cells. It's called interstitial space, right? Between the interstitial spaces of each cardiomyocyte, if something else is accumulating, then that causes the increase in weight and size of the heart. So left ventricular hypertrophy, or in general hypertrophy, is a common term you will see, but there are so many types of hypertrophy. If you read any biological terminologies or the medical dictionary, you will see a lot of hypertrophies like physical hypertrophy, physiological hypertrophy, adaptive hypertrophy, maladaptive hypertrophy. So there are so many types. So eventually, when for a general layman, I would say there are only two types of hypertrophy: adaptive and maladaptive. Adaptive hypertrophy is a healthy hypertrophy, and maladaptive is a pathological hypertrophy, which is not a healthy hypertrophy. So healthy hypertrophy means, for example, sportsmen or athlete, when they run or sprint walkers or sprint runners, so they need, their body have a high demand of oxygen, even though the heart beats a lot. So the pumping of the heart, the blood, because the blood is the one that carries the oxygen, obviously. So the oxygen demand of their muscle for a sportsman or a sprint runner is much higher than the heart, the blood pumps by the heart. So That's why what happens is eventually the heart size thickness increases. So that it's like, if you take a dumbbell and do exercise, your muscle increases, right? That's what a bodybuilder do. It's the same thing. So when the heart pumps more and more, and its muscle increases. So that increase is because of increase in the muscle size or cardiomyocyte size. Like I said, they won't divide. So the only way is they increase the number of the area of cell. So that is adaptive hypertrophy, which is a healthy, and that you can see very commonly in athlete. Maladaptive hypertrophy is not increasing the cell size. It's because of accumulation of collagen. So collagen is also a structural protein. So usually the structural protein of a heart cell or cardiomyocyte is myosin heavy chain. So myosin heavy chain is the very common, highly abundant structural protein found in both skeletal muscle and cardiac muscles, cardiomyocytes. But collagen is present in very minimal, and it is not very required. But the interstitial spaces means the space between the two cardiomyocytes, like in this diagram. So the normal, it's a transverse section of a cardiomyocyte or a heart section. You see that each one is a cardiomyocyte. And this is the fibrotic, means hypertrophic, where the yellow portions are the collagen. So when there is more collagen accumulates between the interstitial spaces, obviously the size and the weight of the heart increases. So fibrotic means, why they call fibrosis means it's like a fiber. So collagen is not a good structural protein. So it becomes stiffens, the cardiomyocytes become stiffens. And when it stiffs, obviously the contractile function will be lost. So even though the size and structure of the heart increases, the contractility of the heart decreases. So it becomes very stiff. So accumulation of collagen is a maladaptive hypertrophy. So which is not a desired hypertrophy. So and the second disadvantage is, because the heart size or the thickness of the wall increases, the capacity or the lumen or the cavity of the ventricle decreases, because it cannot expand indefinitely, right? So there should be a compensation of the increasing of the heart thickness. So the compensation is the ventricular volume goes down. So already the contractile function is gone down. So thereby the pumping capacity is gone down. On the top, if the volume of the heart that can hold the blood is going down further, then there are two impacts on the body. One is the contractile function, the pumping nature of the heart is gone, reduced significantly. On the top, the volume it can hold is reduced significantly. So that eventually cause a lot of cardiac dysfunction, which is called diastolic or systolic dysfunction, and eventually cause myocardial, myocardial injury. So the cardiac cells will die. The other cardiac pathophysiology include myocardial fibrosis, which is, I already explained, the fibrotic, the collagen accumulation. And metabolic dysfunction includes metabolites, how the redox changes, like NADH to NAD levels in the heart. So redox status in the heart cardiomyocytes is very, very important. And then lipotoxicity, which is again the lipid metabolism inside the cardiomyocytes. And of course, this also includes the type 2 and type 1 diabetes, and electrical remodeling. So the contractile or beating of the heart cardiomyocytes is because of the electrical electrophysiology of the cells. So we'll see more in the next slides. So, okay, so what is hyperoxia? Why I'm talking about hyperoxia in this talk, especially for the cardiovascular. So let's see what is hyperoxia, when we need hyperoxia. So the standard, the standard way of treatment for ICU or critical care or emergency department patients, any patient, if they admitted to ICU, the first line of treatment is mechanical ventilation. So whether the patient is preconditioned or not, so usually what they do for few hours to few days, depending on the status of the patients, the first line of treatment is mechanical ventilation. The moment the patient enter into ICU, they give mechanical ventilation. So you must have seen in most of the movies, right? So when they see on the outside of the door, you see ICU, but if somebody peep through the windows and the patient having a mask with oxygen. So that's what mechanical ventilation is. So that is a very common line of administration in ICU. But depending on the condition of the patient, like if they have pulmonary problems or obesity, diabetes, you name it, or cardiovascular conditioning. So depending on the pre-existing condition, the length of stay may vary. So that's only difference. Otherwise, everyone who goes into ICU might be undergoing mechanical ventilation. So what is hyperoxia? Why we need hyperoxia? Okay, mechanical ventilation is fine, we understand. So what is hyperoxia? So okay, let's see why a patient needs mechanical ventilation and what is given in the during mechanical ventilation. So mechanical ventilation is oxygen. So in normal air, oxygen percentage is about 20 to 21%. So in this diagram, a small illustration where the lungs, so one lobe is normal, another lobe is filled with the liquid. So for example, just like a left ventricle I discussed, the lung also have a alveolar space where it holds the air. So the space of the lung that can hold the air is constant, for example. And if the space of the lung, alveolar space, is filled with some pro-inflammatory cytokines or inflammatory cells and other mucus, liquid accumulation, fluid accumulation, the amount or volume of the lung that is sufficient for air to hold, oxygen to hold, is reduced. So just like the heart, so lung also have its capacity. So when there is more liquid accumulation or fluid accumulation inside the lung, then obviously less amount of air it can hold. So the patients that comes to ICU, when most of them may be post-surgery or burns or traumatic brain injury or accidents. So they may have inflammatory cells accumulation in the lung due to the condition, inflammation due to whatever the pre-existing conditions they have. So they may need more oxygen. So the 21% oxygen that is available in air may not be sufficient enough because their lung does not hold normal capacity of the air. So you cannot increase oxygen percentage in the air because it's nature. So only way you can do is pump more oxygen. So how to pump more oxygen is pumping more pure oxygen. So you can pump like 40% oxygen or even 90% oxygen, depends on the patient's condition. So that's what the mechanical ventilation is. So you are pumping more pure oxygen, more than 21%. So 21% is normoxia. Anything more than 21% of oxygen is hyperoxia. So hyperoxia usually measured in a patient. If you see, you would have seen in most of the movies in ICU, a patient is sleeping with a with a mask pumping the air, and in the index finger, one of the finger, there is a clip. So the clip measures SpO2. That's oxygen, saturated oxygen levels. There will be a monitor next to the patient where there will be peaks, the cardiac beating, heartbeat, and also the oxygen levels, SpO2 levels will be shown. So, and also you would have seen in some movies that, some villain come and then he will take out the mask and then the patient die and the peak will become very flattened. So that's what the scenario, that's what the hyperoxia SpO2. So the monitor shows how much oxygen is going into the patient and how the heartbeat is. So higher the oxygen, then even though less volume, because the oxygen is higher volume, higher percentage in the air, so the lung may have enough oxygen to supply throughout the body. So that's the major reason of going for the mechanical ventilation. So leaving it aside, so next, let's go to the next point. So okay, so what happens if more oxygen is pumping? So hypoxia, let me tell you that the terminology, hypoxia and hyperoxia. Hypoxia is less oxygen, hyperoxia is more oxygen. So the patient admitted to ICU have hypoxia, less oxygen in the blood. So they'll be administered to hyperoxia, high oxygen, so that the compensate the requirement. But eventually what happens is the hyperoxia releases free radicals and it causes lung damage into the lung cells. And that in turn causes a lot of inflammatory cell infiltration into the lung and causes more and more lung fluid accumulation, which is called edema. So the pulmonary edema means accumulation of more lung fluid, and eventually the acute lung injury, which is called ALI, acute lung injury, which is a very severe problem under mechanical ventilation patients. So acute lung injury is a well-documented, well-established model in clinical studies. So mostly the physicians try to avoid hypoxia, means less oxygen, because they think patients die, but then ignore how much oxygen they are pumping. So they just start the monitor and then just leave the patient for maybe hours or days, even weeks, depending on the condition. So what happens is every time how much it is pumping, the monitor is recording. So if it is pumping too much of oxygen than the required, so eventually the acute lung injury happens and then the patient dies. So that's why mostly any patient admitted to ICU with mechanical ventilation, if you see the turnover, the outcome, the mortality is very high in those patients. So that's a major problem. So let's go to the relation of hyperoxia with COVID. So we know that, based on the Wuhan and the Italy and other clinical reports, so most of the patients admitted to hospital with COVID-19 are admitted to mechanical ventilation. So why they need mechanical ventilation? So we just saw the first slide, right? SARS. The SARS, the name of the virus itself is a severe acute respiratory syndrome. So because the virus causes lung infiltration of lung fluid, so that induces a lot of lung fluid accumulation. Obviously, they need a respiratory support. So they have a respiratory distress. So obviously, these patients need mechanical ventilation. So 88% of the patients admitted to a hospital with COVID-19 need invasive mechanical ventilation. So invasive means you would have seen in some movies, right? Not the cone, oxygen through the cone, it's through the trachea. So they may put a hole at the throat, or there may be a pipe through the mouth. So there is a small pipe they put through the mouth. So this is called invasive. So this terminology is called intubing, endotracheal intubing. And removing the tube is called extubing. So they put
education sir. Education के बारे में बताइए क्या? Yes sir. Yes sir. No, he's not really good, sir. I don't know how.
Hello.
He's not really good, sir.
Hello. Yeah, I see some voice. Is somebody asking me a question or?
Yes sir. जिसको ये नहीं आता, तो education क्या आएगा?
Okay. Okay, I think they stopped the video. Okay, so the intubing and extubing. So more, so that is called invasive mechanical ventilation. Non-invasive mechanical ventilation is, like I said, through the cone, the mask. They put a cone and a pipe into the oxygen cylinder. So that's non-invasive mechanical ventilation. So the very less percentage of patients with COVID-19 require ended up with non-invasive 11%, but major chunk of the patient population requires invasive mechanical ventilation. And also the data shows that 79% of the patients that require that had non-invasive mechanical ventilation had a mortality, whereas 86%, which is very high, mortality is found are recorded in invasive mechanical ventilation. So that clearly says that invasive mechanical ventilation have higher mortality than non-invasive. Of course, both have high mortality anyway for COVID-19 patients. So that is a direct association of hyperoxia with COVID-19. Our lab is the pioneer in studying how the hyperoxia remodel the cardiac physiology, both structure and function. So we are the pioneers. We started this study almost 7 to 8 years ago, even before COVID-19 happened. So according to CDC, we know that the patients admitted to hospital with COVID infection, higher chunk percentage are like 75, close to 75% are older adults, like 50 to 60, more than 50 years old. So geriatric population. So meaning that the older is a patient, higher is the chances of infection. At the same time, higher the incidence of mechanical ventilation. So the age is directly proportional to the mortality. So that's the second factor. So we started in animal experiments that for 3 days of hyperoxia. So we use mice model, and we started 72 hours of hyperoxia treatment using a wild mice model. So the wild mice means meaning that these mice are healthy. So these are not COVID infected. So we started with just healthy mice. We want to know we want to know the effect of hyperoxia alone without any other comorbid conditions, without interference of any other risk factors that can interfere our data. So that's the reason we took healthy mice first. So obviously in clinical practice, it may not be healthy human admitted to ICU. So that you can imagine by after looking at our data of healthy mice, you can imagine how the patient with other conditions would be. It would be even severe. So what we found is most of these mice admitted to hyperoxia for 72 hours or 3 days have left ventricular hypertrophy, ion channel remodeling, and arrhythmia. So ion channels means that the heart cells require sodium, potassium, and calcium. These are 3 major ion channels that that are responsible for cardiac electrophysiology. So because of the electrophysiology of the heart cells, the cardiac contractility or pumping of the heart, beating of the heart is possible. So we'll see more into the hyperoxia, how we did the hyperoxia experiment. Okay, so this is the chamber, the hyperoxia chamber, because I cannot give mechanical ventilation at a pipe into the mice because, you know, human is different. They will be under anesthetic or they can be, you know, put down on the bed and do it. So animals are difficult. So they tend to move freely. So what we do is we have a chamber, a airtight chamber with oxygen sensors, inlet and outlets, and with the shelves, and each shelf carries 2 to 3 cages. So each cage, again, we can put up to 4 mice. So we put mice cages inside the chamber, airtight chamber, and then we pump 80 to 90% oxygen, pure oxygen, and sometimes even 100% oxygen. Anything above 70% oxygen is fine. We keep that, usually we take mice of age 8 to 9 weeks, which is almost adult age, and then expose for 3 days, and then we stop the hyperoxia. So another group of mice of same age, we try to take the same weights, body weights initially. So some mice, we group them as hyperoxia. Another group will be normoxia. Normoxia means they will breathe normal air, no oxygen, just normal air. And then we compare between hyperoxia and normoxia. So when we compare the physical parameters like body weight, so even though all these mice, both normal air and hyperoxia treated mice, they started with the same body weights. After 3 days of treatment, if you see the panel A, the body weights of normoxia is normal. It remains constant, similar, but the body weights of hyperoxia mice, even though they started with the same weight of normoxia, normal air mice, but they go down very, very significantly. So they reduce body weight. This may be due to the pulmonary sickness, pulmonary distress. So they may not eating much, they may not drink much water. So probably the eating and drinking habit might have gone down, but the total metabolism go down very, very drastically. So their body weight go down very significantly. But if you see the heart, usually the body weight and heart proportion are very proportionate to each other. When the body weight go down, we imagine that the heart also go down, the size and weight of the heart go down, but it's not true. So if you see the panel B, the physical image of the entire heart, so you can see that normoxia heart, normal heart, how it looks like, and the hyperoxia heart, very huge and large, too big. The weight, when I checked the weight, I thought it can be possible that the size may be increased, the weight may be same because of stretching of the cells. So, but that's why I weigh the hearts also, but I saw the heart weight also increase very significantly. So it's not only the size, but also weight of the heart. So it means there is something adding to the heart like collagen. So I was imagining that, so there may be highly fibrotic, it's undergoing fibrosis, maladaptive hypertrophy. So that's the reason maybe the heart weight also increase. So when I check the heart cross sections, so you can see that normoxy heart here and the hyperoxy heart here. So this is the right ventricle and this is left ventricle. You can see that the thickness of the septum is very much increased and the left ventricle is very thick. And the area, this is the area of heart section. So hyperoxia significantly increase the heart weight and also heart size and thickness of the valves, each cardiac valve. Then we examine the serum markers. So like I introduced that LDH, lactase dehydrogenase, and the troponin, cardiac troponin, are two markers that usually any cardiologist measure to see if they have cardiac problem. And we found that both LDH and troponin are very significantly elevated in hyperoxia mice. So that is an indication that they are having a severe or very high cardiac injury. And then we also did surface ECG, electrophysiology, electrocardiogram, and we found arrhythmias. So you can see that in this panel, you see that each peak is 1 beat, heartbeat, if you say. So each peak, you say that this is a beat. The distance between these two beats is the time it takes for each beat. So they are very rhythmic, so very uniform. But if you see the hyperoxia, these distances are different, and then there is a gap, and then after this panel, there may be less distance, and then far distance and medium distance. So there is no rhythm, there is no uniform distance between the peaks. That's called arrhythmia. So hyperoxia mice showed a clear arrhythmia, and when I dissected each peak and then further analyzed, there is a QT prolongation. So Bojbabu, I think he would have given you some introduction of how to measure QT length. So this is the further dissection of each peak where I can clearly show that QT prolongation happening. So this is the mean value of all the data, which clearly says the QT prolongation. So in addition to the ECG, what we did is there is an experiment called ex vivo, where after we anesthetize the mice and we cut the heart from the mice. When the heart is beating within few seconds, maybe in fraction of seconds, immediately we had to mount the heart into equipment, Langendorf equipment, where the heart is still beating, and we pump the solutions through a pump, mechanical pump, so to make the heart beat, and all the solution, the buffer have all the ions that is required for the cardiomyocytes to beat. And then with the help of electrode, this is an electrode, with the help of electrode, we measure electrophysiology of the heart, beating heart. So here in this apparatus, I'll show you the pointer. So here in this video, you can see that how the heart is hanged to the cannula and then run through the pump. You can see the heart is beating, and then we are recording through the electrode. So this is called Langendorf equipment. So we use this equipment to see ischemia reperfusion. Ischemia reperfusion means in humans, sometimes the heart pumping the blood stops. So there is through maybe a blocking, because of a blocking of the blood vessels or something, we can say the circulation of the blood from heart to the organ will be stopped. So it can be a stroke or heart attack. So that the pumping is stopped for it may be for half an hour or 1 hour, for some time. So then the clot may go down, or the blood vessel may dilate further to ensure the blood flow. So the blood flow may come back. That's called reperfusion. Ischemia is stopping the blood. Reperfusion is pumping the blood back. So we mechanically did that. We stopped the perfusion of the liquid for 30 minutes, 15 minutes to 30 minutes, sometimes even 1 hour, and then pump the blood back and see how the electrophysiology of the heart happening. So here, if you see that, so the panel A is electrical peaks, repolation peaks of normal heart when it is no ischemia, no stopping of the perfusates, no stopping of the buffer with the continuous flow of the buffer. the heart is, the peaks are like this. But after ischemia of 15 to 30 minutes, you see that peaks are like prolonged. So this is called action potential prolongation, APDs. So action potential duration is prolonged. So I'm not going too much detail into electrophysiology. I know it will be very boring and confusing. So I want to just give the, the synopsis or the main essence of what is electrophysiology, what I'm looking for. So there is a clear delay in the action potential duration. So the solid line in D panel, you see that each peak, the duration from 50 milliseconds, and but in the ischemia heart, it is prolonged further to come back to the baseline. So that clearly indicates that the hyperoxia caused a lot of action potential delays. So we know that a lot of literature says that potassium channels, KV channels, we call commonly, involve in action potential duration of each cardiomyocyte. So I examined at molecular level. I want to know that what is making this cardiac dysfunction. So what genes are actually involved in that? So based on the literature, potassium channels play a major role. So I studied real-time PCR and Western blots of a lot of KV channels, KV 4.2, 2.1, 4.3, and their KV channel associated genes, and most of them, some of them downregulated, some of them upregulated in hyperoxia, and MSC 6 and 7 are myosin heavy chain. Like I said in the introduction, myosin heavy chain is the major structural protein of skeletal muscle and cardiomyocytes. So these also upregulated a lot, meaning that the size of the cell is increasing. That's why the hypertrophy is. So taking the summary, stopping here, I have a lot of data, tons of data. I don't want to make you too much confused with a lot of cardiovascular, cardiac physiology. I just want to give very important things that a normal, a common man can understand. So until now, the summary is the data clearly shows that hyperoxia can induce cardiac dysfunction via increasing heart rate and also ventricular hypertrophy. And also it can cause cardiac injury, which is evident by elevation of cardiac markers like troponin and LDH. And electrical disturbances or electrophysiology, dysfunction causing arrhythmia and other KV channel dysregulation. So these are some of the reasons how the hyperoxia can induce cardiac dysfunction. So now comes to the question that we did all these experiments in the male mice because female have hormonal influence, estrogen influence due to the estrous cycle. And there is a lot of literature says that clinical, both in human and animal, says that there are gender disparities in cardiac functioning in both men and women. So I want to see that is it true in case of hyperoxia also? Is it because we know that the heartbeat, heart rate in female is higher than male, and also their size is very small, heart size is smaller in female. That's very normal. And their QTC, female already have a QTC prolongation. Like I showed in the hyperoxia mice, how the QTC prolonged. The normal woman in normal healthy condition itself, they have QTC prolongation. So the females are entirely different than males. So I want to see that, is it true with the hyperoxia? But there is some misconception previously that women are highly protected from cardiovascular disease. If you see the textbooks or physiology, cardiology books, in older books, older versions, you might see that women are more protected from cardiovascular disease than men, but that's not true. If you see CDC data over the past few decades, there is an equal incidence of cardiovascular disease in both men and women. Obviously, with no doubt, estrogen have some protective role, but not in case of hyperoxia. I will show you that how the hyperoxia, how the female behaves. So here in this experiment, you see that both male and female are compared for 3 days of hyperoxia. I took the same age groups because I cannot take the same weight between male and female because female mice always smaller and way lesser than male. So I took the equal age. Age is as constant factor. So equal age mice, male, one group undergone normal air, one group hyperoxia, same thing female, normal air and hyperoxia. So just like any male and female. So I can clearly see that hyperoxia reduce body weight, just like we observed in the male. So there is no difference in the physical parameter in the female. But heart rates, surprisingly, like I showed in the previous slides, the hyperoxia, mechanical ventilation, increases the heart rate in male, but not in female. So that's a surprise. If you see in the panel D, the male heart are increased after hyperoxia treatment, but female hearts are not increased. If you see the cross section, in fact, the female hearts are getting much smaller than the normal heart. So that is a significant difference in female than male in physical parameters. So next, going to the echocardiogram. This time I did echocardiogram also in addition to ECG, electrocardiogram. So echocardiogram shows the function of the heart, how the heart is pumping, systolic and diastolic function. So that shows that both male and female have a very severe cardiac dysfunction. I am not going too much detail. If anybody who is good in echocardiogram, they can see the data here. Left ventricular diastolic systolic function, and end diastolic volume, end systolic volume, and stroke volume, cardiac output. So these are the measures of cardiac function. So both male and female after hyperoxia have very severe cardiac dysfunction. And just like male, even the female mice also shows arrhythmia after hyperoxia treatment. Like I said, the female already have QTc prolongation even at normal air, but after hyperoxia, that even exaggerates. That's even prolonged too much. So that's going to be pathological, even severe. So another thing what I observed is these female mice have started having 50% mortality. So I never had any male mice died during 3 days of hyperoxia, but when I did the same experiment of 3 days hyperoxia with the female mice, 50% of my mice died. So that clearly shows that females have higher pathological problems, cardiopathology, pathophysiology than the male. So of course, the same panel, same like male mice, I see all the potassium channels dysregulation in both male and female and the hyperoxia. So taken together, the overall summary is just like a male, the female mice also have arrhythmias, cardiac dysfunction, and defects in cardiac repolarization reserves and KV channel remodeling. So these things are constant, are same between male and female. So what is significant or different in female is the females have smaller body weights and smaller hearts, of course, and they have prolonged QTc, which is a precondition in the female. It's also observed in human, not only in mice, even in normal air, even before hyperoxia. But when they exposed to hyperoxia, the QTc prolongation is further prolonged, even worse, and they undergo bradycardia. Bradycardia means reduced heartbeat. So the heartbeat goes down under hyperoxia, and I see 50% mortality, and there is no hypertrophy. So no increase in heart rate, just like we observed in male, there is a hypertrophy. In female, we didn't see hypertrophy under hyperoxia. So these are the differences between male and female mice model, healthy mice model under hyperoxia. So now come to the point that you may have a question that why we are discussing all this, why not on COVID? What is its relation to the COVID? Okay, let's see that how I can convince you that hyperoxia is the one that caused a problem to the COVID patient, not the viral infection. Is if you see the top panel, the X-ray of a healthy normal patient and acute lung injury due to hyperoxia, hyperoxia-induced acute lung injury patient. So there is a lot of smoky appearance. This is because of the accumulation of lung fluid. So you see that a clear lung versus the fluid-filled lungs. So same thing, exactly same thing, but only thing is the way they took the diagnosis is different. Here on the top panel it's X-ray, here it is MRI. So in the radiology diagnosis, if you see the down panel, the normal lungs versus the COVID-infected lungs. So there is a lot of inflammation or fluid accumulation in the lungs. So very, very similar lung physiology in hyperoxia-induced acute lung injury patient versus the COVID patient. So which is highly convincing for me that it's not the viral infection, it's actually the hyperoxia or mechanical ventilation that is causing death to these patients. So then my third question is, does age have any role in hyperoxia-induced mortality? Because we see that a lot of data from WHO and CDC saying that age is a risk factor for COVID infection, and the aged people, if they are infected with COVID, there is a high mortality than the young adults. So I want to see that, is it again because of hyperoxia? Because hyperoxia have age differences, or hyperoxia-induced cardiac remodeling is influenced by age. If that's the case, then obviously the hyperoxia is the one that is causing mortality in the aged population also. So then what I did is I took healthy, normal aged mice of 58 weeks, so which is equivalent to more than 50 years of human age equivalence. And then I exposed them to 3 days hyperoxia. You can see that similar to the any data I have published, the body weight significantly go down and the lung dry to wet weight. So the reason why I took lung dry to wet weight ratio is lung weight, wet weight I take, and then I dry the lung for 2 days to 3 days at 50 to 60 degrees centigrade. So all the liquid, it is there, it will be dried and evaporated, and then we weigh the lung. So if it is still heavy, it means there is a lot of accumulation of other lung fluids or other fibrosis. So that's what the ratio is. So there is a significant increase in lung wet weight to dry weight ratio. So it means that the lungs are getting a lot of lung fluid accumulation in age under hyperoxia without any COVID infection. So just hyperoxia is filling the lungs with a lot of fluid. Then I saw that same electrophysiology, ECG. So there is a QTC prolongation and other PR and other electrical prolongations, which is evident by ECG, surface ECG. So overall, the summary is saying that so that 50% of the population admitted to ICU in general, and not only COVID infected patients. So general statistics, if you see the data from any clinical reports, you can clearly find that 50% of the population admitted to ICU undergone mechanical ventilation are more than 60 years of age. So they're all geriatric population. So more the age, more chances they end up in ICU. And outcome of the patients from ICU with the age is very less. So more the age, high is the mortality. So there is a very less chances that they come out of ICU and discharge. So when there is a higher age, high incidence of mortality. And also the census, the report says that by 2030, 20% of the world population will be older adults, and definitely cardiovascular disease will be a significant problem going forward. And we saw that our data clearly shows that similar to young adult mice, the aged mice also have significant weight loss and lot of accumulation of lung fluid under hyperoxia. The age mice, even at normal air, have prolonged QTCs, which is further exaggerated under hyperoxia, and looks like they may have high mortality than the normal young adult mice. So that clearly says that age itself have some direct influence on hyperoxia-induced cardiovascular changes. On the top, if these mice have COVID infection, you can imagine how severe. How I am saying is from the data, clinical data from Wuhan and Italy, and they have clearly shows that the patients, the hospitalized patients with COVID infection, 74% are geriatric. Out of that, 54% are male. So there is a high percentage, like almost 75%, 3/4 of the population admitted to hospital with COVID infection are aged population. So aging is a very, very severe influencing factor for COVID and the cardiovascular, and of course the mortality. So the mechanical ventilation is another factor that impact the cardiovascular disease or mortality rate. And imagine if these all the independent risk factor, for example, diabetes itself is one cardiovascular independent risk factor. That is a well-known phenomenon. And aging, we know that the diabetes, incidence of diabetes or prevalence of diabetes increases with age. So if the aged person with diabetes admitted to hyperoxia and having COVID infection, so it is like 4 independent risk factors having on the same patient. You can imagine how much the mortality is. Maybe very, very close to 90%. There is no wonder. This, if the patient having this kind of triple or 4 independent risk factors together, then it will be severe. So going forward, so what is the overall summary I want to say is that it is hyperoxia that can cause severe lung and cardiac injury. It's not COVID infection. Of course, COVID infection has some effect on lung. That's why they are called SARS. So they have some lung fluid accumulation. But if the patient is admitted to mechanical ventilation for a long, prolonged period without regulating the oxygen, percentage of oxygen, this patient eventually become ALI, acute lung injury patient, and then there is a high chances that the patient may not return back. On the second point is the age. Age is an independent risk factor. So higher the age, more chances of mortality and less chances of returning back from the hospital. And then the gender disparities. Of course, female and male have entirely different significance with hyperoxia, and female shows higher incidence of cardiac injury than the male. So we don't know with the COVID. I'm currently working on that, the gender disparities in COVID infected mice under hyperoxia. I'm working on it. And also I'm working on different aspects like estrogen. If I inject estrogen, because the older post-menopause, we can say after 50, the post-menopause women have high cardiovascular incidence because they lack of estrogen. So can the estrogen therapy can improve? If so, if the woman admitted to ICU under mechanical ventilation, even with a normal estrous cycle, if we inject estrogen, can the estrogen protect the heart from hyperoxia-induced cardiac remodeling? So that also I'm working on it, hormonal therapy, both estrogen and androgen in male. So I have some data. It's a tons of data. I don't want to confuse you with too much of cardiovascular physiology. And the second take-home point is that so the physicians should look for alternative approaches especially for COVID patients with high age or older age. When they are admitted, of course, they need oxygen supplement. So give them intermittent oxygens at few hours, and then normoxia, and then few hours of hyperoxia. So intermittent oxygen supplement instead of continuous supply without monitoring it may improve the outcome from the ICU. Or there are some drugs that can dilate the alveolar space or lung that can improve the lung capacity. So using some drugs, or if possible, avoiding hyperoxia totally can improve the survival rate for these patients. So this is the take-home message. I don't want to impact you with tons of data. I have a lot of data, so it's all physiology and electrophysiology and molecular biology. So I know it's a long day for you. You have been listening very patiently on too many lectures and too much of science. Let's discuss so far what we have. If anything related to cardiovascular COVID, I'm happy to answer you.
Sir, thank you very much for your presentation, sir. One question, sir.
Yeah.
How much risk to asthma patients compared to normal patients?
Oh yeah, they are. I forgot to mention that. So there are two conditions called COPD and ARDS. So COPD means chronic obstructive pulmonary disorders. So which includes bunch of pulmonary disorders like asthma, and pulmonary hypertrophy, fibrosis, and smoking. These are all included under COPD. And same thing, ARDS, acute respiratory distress syndrome. So the patients with these pre-existing conditions definitely need hyperoxia. So they will die without that if they are infected with COVID, because they already have a pre-existing lung condition. On the top, the COVID induce more and more lung fluid accumulation. So they will have a very severe impact irrespective of their age. So these patients should be monitored very closely about hyperoxia, how much oxygen they are pumping. I know that it's very impossible for a physician to go with an armoxia. They have to go with hyperoxia because of their lung problem, but not too much of hyperoxia. So they, like I suggested, that intermittent oxygen increase and titrations so may improve, and close monitoring for these patients at least is very much required.
Thank you very much, sir. And only one question, sir, for you. That's it. Thank you very much, sir. And in the chat box, all are appreciating about your presentation, sir. Thank you for sparing your time and delivering the informative message to all the participants. We look forward for in future, sir, for your collaborative research work. Thank you so much.
Sure. Yeah. Thank you very much.
Thank you. Now I invite the principal, College of Science and Technology, Dr. K. Ramaneshwari, to deliver the concluding remarks. Madam.
Thank you.
Hello.
Yeah, I'm there online.
Okay, madam.
Thank you, Dr. Vijay, for asking me to deliver the concluding remarks. And as it is delayed session, I would like to conclude very quickly. Hope and faith is what keeps man going every day, even though how difficult the situation is. And as the COVID-19 pandemic continues, the researchers and the manufacturers are moving potential therapeutics into clinical trials at a very rapid and busy pace. And as we have seen from our presentations, from our resource persons, the eminent speakers, their studies, the structural biological studies, the molecular biological studies, the physiological studies, the genomic studies, in addition, are contributing towards this hope and faith, so that research continues to find the therapeutics and also the vaccine. And this is potential therapies are being examined in several countries. And the largest one is the group Solidarity, that is being monitored by the WHO, which includes about 100 countries to evaluate the high-profile treatment candidates for COVID-19. And if you see the response from the pharma and the biotech companies globally in finding a COVID-19 vaccine and the therapeutics, we can see that I have found these statistics on the internet. There are about 438 unique drugs that are being tested, then 298 therapeutics, and are about 140 prophylactic vaccines, which are in different stages of development, like discovery, preclinical, the phase 1, phase 2, and phase 3. So this massive contribution, it has resulted in some of the drugs being tested, like those which have been mentioned by the speakers, like the HCQ, then remdesivir, then favipiravir, and the other hope-giving vaccine that is from the Moderna labs. I think this is what you say, a massive pipeline in such a short time is the demonstrative effort of the pharmaceutical industry, which is because it has compiled the resources and the talent of finding a drug to combat this pandemic. And to find a drug, only the pharmacy manufacturers are not important, but all the knowledge that is coming out from the scientists, the researchers, the academicians is important in doing so. I think this is the first step in contributing to fight this COVID pandemic. And with this, I also thank all the resource persons, and especially I am proud of my student, Dr. Ruchi Babu, who has been on the list of the speakers. And I once again thank the convener and congratulate the department for conducting this webinar. Thank you very much.
Thank you very much, madam, for your concluding remarks, very brief and detailed remarks. On behalf of our university, I take the privilege of thanking the speakers, Dr. Tangamalai sir, Dr. Bujji Babu, Dr. P. Gopinath, and Dr. P. Sivakumar. Once again, thank you for your excellent presentations. I thank the participants, my colleagues, faculty friends for their support, and the participants for participating. Thank you one and all. The session is ended.